sit down. A very good morning, ladies and gentlemen. I, Diksha Shinde, welcome you all to the second day of 2 days international conference on advances in pharmaceutical science research, traditional to novel approaches, organized by Dattakala Shikshan Sanstha's Institute of Pharmaceutical Science and Research for Girls College, Swami Chincholi, Maharashtra, in association with Dr. Babasaheb Ambedkar Technological University, Loneri, Association of Pharmaceutical Teachers of India, APP, APTI Maharashtra, and Association of Pharmacy Professional, APP Maharashtra. I would like to welcome our resource person, respected Dr. Haider Al-Musawi sir, Dr. Jokha Al-Kasabi ma'am, and respected Dr. Muhammad Ishaq sir. Please welcome all of them. I also welcome all the respected dignitaries present over here, respected Dr. Vishal Babar sir, Chief Administrative Officer of Dattakala Shikshan Sanstha and Principal of IOPSR. I would also like to welcome respected Jyoti Zavle ma'am, Vice Principal of IOPSR. I would also like to welcome all the principals from various colleges present over here. Lastly, I welcome all the participants from various colleges all over India. Additionally, I would like to extend a hearty welcome to all the dignitaries. Yeah. For the first day of international conference, we have very knowledgeable session. Keeping this momentum, we will proceed to our third lecture session of the second day of international conference. At this moment, I believe we are fortunate to have such distinguished knowledgeable speaker, and I promise that these sessions will be beneficial to all of you. So I request Alpha ma'am to introduce our first speaker, Dr. Haider Al-Musawi sir.
Good morning everyone. I, Miss Alpha Jain, welcome you all on this 2-day international conference which is held at Dattakala Shikshan Sanstha, Institute of Pharmaceutical Science and Research. Respected Dr. Haider Al-Musawi sir, welcome sir. Sir is currently working as Faculty of Pharmacy and Pharmacy Expert at Muscat, Oman College of Health Science, Oman. Sir has completed Master of Pharmacy from University of Stradgland, Glasgow, United Kingdom in 2011. Sir has also completed Master of Science in Clinical Pharmacy from Robert Garden University in 2017. Sir has completed PhD in Molecular Pharmacology from University of Stradgland, Glasgow, United Kingdom in 2024. Sir is having more than 15 years of research and teaching experience with various expertise in the subjects like pharmacology, pharmacy practices, clinical training, dropping in sports, etc. Sir's research focuses mainly on molecular pharmacology, cancer treatment, pharmacotherapy with experience in various laboratory techniques which include Western blotting, cell culture, EMSA, assays, etc. Sir has delivered various lectures in national and international conferences, guest lectures, which includes Ibra Hospital's second Pharmacy Symposium. Sir has a speaker in different professional development programs. Along with that, Sir has received third award in poster competition, which is arranged by Omani Student Advisory Councils. Along with that, academics, sir is also having different experiences which include clinical pharmacist at the NICU, Kola Hospital, which includes patient counseling, prescription management, physician advisement on the drug dosing. Sir has delivered various talks in the national and international conferences. Sir's works glioblastoma research, which leads to discovery of the novel IIK selective inhibitor, which get patented. So we are eagerly waiting to listen to sir, which is sir is actually delivering on the same topic, which get patented, sir. That is talk on the adverse pharmacological strategies in the glioblastoma management targeting the IIK signaling molecule for the novel drug discovery. So I hand over the session to you, sir. Thank you.
Still doesn't work? Does it work? Okay. Do you hear me well? Good. This is a point.
Pointer.
And changing the slides. Up and down. Okay. This is private probably. Okay. Good. Okay. Right. Good morning. Thank you very much. Before I start my presentation, I would like to say thank you from my heart to all India, for they are so welcoming, for the very nice welcoming and hospitality and the beautiful culture that we've already experienced so far, even the spicy food, which I'm not a big fan of, but I tried it. And this actually on behalf of all entire man. Thank you very much again.
Also like the respectful attitude of these
Students. So big clap for them. And I wish you a very best in your career, and I wish that one day you all be the greatest pharmacist in the world. Shall we start now? Yep. So, my talk today is going to be about brain cancer. Whenever we talk about cancer, it's always a complicated subject. But I'm going to try my best to make it as simple as just drinking water. Hopefully. So to start with, what do we mean by cancer? Cancer is just cells that transformed to be abnormal. So we have normal cells with normal characteristics, and we have abnormal cells with abnormal characteristics. And that's cancer, simply. And brain cancer, I chose brain cancer because it's the most aggressive type of cancer. And specifically speaking, the 4th grade of brain cancer, and it's called glioblastoma multiforme. Glioblastoma G, start with G. So G, glioblastoma B, and multiforme M. So GBM. You will hear me saying GBM the entire presentation. So GBM is glioblastoma multiforme, which is a stage 4 brain cancer, the most aggressive type of cancer ever. The mean survival period of a patient with GBM does not exceed 15 months only. That is with the current therapy approach, which consists of chemotherapy, temozolomide mainly, surgery, debulking surgery, and radiation therapy. We don't have anything else for GBM management, unfortunately. Until today, there's no cure for GBM. And that's why I studied GBM, and I was very keen to come up with a new management strategy or a new drug that might cure the disease, hopefully, or at least increase the survival period with a better quality of life. That's why I chose brain cancer. And at the end, we will see the results that I came up with. Some prevalences. Cancer, unfortunately, is actually increasing in number. So the number of cases of cancer are actually increasing, even in the future. That's the bad news. But the good news are that the number of deaths, of premature deaths due to cancer, are actually decreasing. And I can only attribute this to the fact that we are advancing in technology in the diagnosis of cancer, and we are also revolutionizing the management of cancer. So we will have increasing number of cancer, but we will have better management of cancer as well. The current treatment options of cancer. What do we have in hand now? Mainly surgery, chemotherapy, radiation therapy, hormonal therapy, and a few touches here and there in immunotherapy, shy in immunotherapy, targeted therapy as well. Bone marrow transplantation is very specific to certain types of cancer like leukemia only. Right? However, can we do any better? And that's what I'm going to talk about today. My name is Haidar Al-Musawi. Thank you for your introduction. That was a brilliant introduction, more than brilliant. My name is Haidar Al-Musawi. I'm an assistant professor of pharmacology from Amman College of Health Sciences. My PhD was mainly concerned about brain cancer. So I'm going to talk about the advanced pharmacological strategies of GBM management, specifically talking about targeting IKK alpha signaling in GBM. It sounds difficult, but I'm going to simplify it. It's interesting. Just pay me your attention, please. To start with cancer, cancer has different characteristics, as I said, than normal cells. So what are these characteristics of cancer that makes it a bit different than the normal cells? Well, first thing is sustaining proliferation. Cancer cells can proliferate and differentiate continuously. Normal cells, they don't. So, a stem cell that needs to be differentiated to stomach cells, it will be stomach cells. That's it. In cancer, no. A prostate cancer cell can start as a prostate cancer cell, but then it might migrate somewhere else in the body and become bone cancer cells. So it keeps proliferating and differentiating as long as it moves in the body. That's one. Evading growth suppressors. Normal cells actually in our body have gene suppressors, growth suppressors. So it plays like a red signal for the growth of the cells. So back to the stomach cells. If we manage to form the stomach, then there is a stopping gene says no more growth is required. That's enough. Now cancer cells can evade these suppressor genes and keep growing and growing and growing and forming tumors, what we call tumors. Avoiding immune destruction. This is quite new actually. Recently the scientists found that one of the best fighters of cancer in our body is our immune system. So they started to use what we call now immunotherapy. The entire idea of immunotherapy is increase the immune system of the body to fight cancer by different means. We will come to that in a minute. So cancer cells have the ability to avoid immune system destruction, enabling replicative immortality. Cancer cells are immortile. They keep replicating and replicating and replicating non-stop. Normal cells are motile, and the enzyme that is responsible about making our cells motile or immortile is an enzyme called telomerase enzyme. After each cell division, this telomerase decreases until the end, until it depletes. There's no more telomeres in these cells' DNA. So no further replication can be happening. In cancer, there's a gene expression called human TERT that keep increasing and forming telomeres after each replication. So the cells become immortile, keep replicating non-stop. Activating invasion and metastasis. So cancer cells can invade nearby cells or can even migrate to other cells and cause metastasis. So they can metastasize to other faraway cells. Induction or inducing angiogenesis. Angiogenesis is a term that means forming new blood vessels. So cancer cells can keep forming new blood vessels to supply it with enough blood and nutrients to keep growing, because it needs more blood supply. Resisting cell death, which is what we call normal or programmed cell death, apoptosis. So cancer cells can avoid apoptosis. Deregulating cellular energetics. So cancer cells can use different kind of energy, not only like glucose or maybe oxygen. They can also utilize other things like lactose, for example, as a source of energy. So these are the 8 very well-known cancer cell characteristics. They now call them hallmarks. And anyone interested about cancer will come across hallmarks, cancer cell hallmarks, a lot. These are 8. There are about 6 more suggested coming in the future, probably. 6 more different hallmarks. Why it is important? Because if we understand how cancer evolve and survive, we can interfere in some of these steps and destroy cancer. Okay? So understanding the physiology of cancer cells allow us to understand how can we manage cancer cells. So what is the future cancer treatment? What is coming in the pipeline? There are things that actually have been already released in the market. There are things still in the pipeline. Let's see. Mainly targeted drug therapy, number 1. And that's my topic today. I'm going to talk about targeted drug therapy. Immunotherapy, there's an evolving science behind the immunotherapy nowadays. Cell-based therapy, epigenetic drugs, angiogenic drugs, and suicide gene therapy. Starting with angiogenic drugs. Now we understand that angiogenesis is the formation of new blood vessels. If we manage to block the factor responsible about forming new blood vessels, we will block angiogenesis. We will cut the blood supply from cancer cells and kill cancer cells. So the factor responsible about forming new blood vessel is called vascular endothelial growth factor. So we block this factor and then we will inhibit cancer angiogenesis. Some of the drugs like bevacizumab is actually newly released and is approved by the FDA last year to be used in the management of GBM. The only drug in the past was temozolomide. Now we have bevacizumab, which is a monoclonal antibody. Until today, it hasn't been approved in Europe yet, but it is approved in the UK, in the US. Suicide gene therapy, the idea is that we introduce a gene with a prodrug. This gene is very specific to the cancer cell. It goes inside the cancer cell and it releases the prodrug. Then the prodrug will get activated and kill the cell with the gene. That's why it's called suicide gene. Examples we have things like cytosine deaminase, the gene, the suicide gene, with 5-fluorocytosine, the prodrug. Or maybe we have something else like herpes simplex virus gene with ganciclovir as a prodrug. Other ideas also available, like silencing RNA. How about cell-based therapy? Cell-based therapy is actually very evolving nowadays, even in India, by the way. The idea of cell-based therapy is simply we take cells from the patient, we re-engineer them, and we put them back to the patient. What is behind that? Let's see. Now, the very well known is the CAR T cell-based therapy. CAR T, CAR is the abbreviation of chimeric antigen receptor. So the thing is, as I said, that cancer can avoid the immune destruction by removing the sensitivity to the antibody interaction. So what we tend to do, we take the T cells from the patient and then we introduce an antigen receptor on it and then we give it back to the patient. Now we put a flag on the T cells. This flag will allow the T cells to interact with the cancer cell again. We'll find the antigen and we'll interact with it and then it will kill it. The thing is with the CAR T cells, it's better not to be used alone. It has to be given with other chemotherapeutic agents. It's very newly used, even in Oman, but there are some concerns about it. One of which is the expense. It's so expensive. So expensive. One single patient treated with CAR T therapy requires about $500,000 each patient. However, there are now some initiatives of reducing the price. One of the initiatives actually comes from India. They managed to reduce the price by 10-folds. So about $50,000 with a high quality CAR T therapy. That's good. I actually learned this a few weeks ago from scientists from India. The other limitation to CAR T cell therapy is that the patient has to have certain number of T cells available in his body. Less than that number, it won't be any effective. So patients with immunocompromised patients, for example, are very hard to be accepted to use CAR T cell therapy. Epigenetic changes. What do you mean by epigenetic changes? These are changes in the genes that form wrong DNA. And this mistake can cause cancer. So what we tend to do, we use epigenetic drugs that can correct these changes and treat cancer. This approach is still in the pipeline. And some drugs have been approved so far, like decitabine and vorinostat. These are just newly approved to be used in cancer, but they are not used alone because the profile of these drugs is not fully understood. Targeted drug therapy, the thing that I'm going to concentrate on, the idea of targeted drug therapy is that we try to target very specific signaling pathways within cancer cells. And we block this pathway. Specifically, we block something that is rate limiting within the pathway. Once we block it, we will stop important signals to pass to the cancer cells and make them for example, proliferate or maybe become immotile. We will see some examples. So as I said, we block this channel, this signaling pathway, and then we will kill cancer. Targeted drug therapy is mainly concerned by molecular pharmacology. And yesterday Dr. Ishaq talked about this in a bit. These are some pictures of me in the lab. I was so much enjoying working in the lab. I'm giving these informations, I know it's quite scientific, but maybe someone here in the room might be enthusiastic to do some scientific, basic science in the future. One of these students, it was me. In fact, my M-Pharm project supervisor, he was the same supervisor in my PhD. I loved my M-Pharm project so much that when in the future I wanted to do my PhD, I said I will never do it except with this supervisor, Andrew Paul from Strathclyde. So I went back to Strathclyde, I met him again, he remembered me well. He said, I didn't realize that we will come back again. I said, I love the project, I love basic science, and I want to do it again in full extent. So I've done my PhD with the same supervisor. So molecular pharmacology actually plays a very important role in what we call network analysis and dynamic modeling with the chemistry guys. So we try to analyze the network within the cell, and then we try to find out very important signals that we might block by some drugs, some agents that can enter the cell and work on it. So small molecules. Then we try with the chemistry guys, we sit with them, we say, well, we want to block this biomarker here. Then they will formulate something for us. They will give it like a gift. Try it. And then we try it, we'll see what happens. If blocking this biomarker give a nice effect, then we will carry on. If it causes side effect, then we might do some remodeling and change it a bit. We will sit with them again. Well, we've seen some side effects. Can you try different? Can you change it a little bit? They do the dynamic modeling. So there's a nice communication between the pharmacology or molecular pharmacology section and the chemistry section. And I mean by network analysis, this is one of the networks that happens within a cell line, within a cell. So many kind of signaling pathways. They are interconnected, they communicate with each other. So it's not easy to find a rate limiting step to block. We might block one of the biomarkers, but blocking one biomarker might lead to blocking other signaling pathways as well. So this might lead to either good effect or side effects. And you've already seen this yesterday with Dr. Ishaq, some of the dynamic modeling in CDD. So targeting IKK alpha signaling in GBM cells, that was my concern. I was trying to find a very important signaling pathway within a cancer cell, very predominant in almost cancer cells, almost all different types of cancers. And that was what we call the nuclear factor kappa B pathway, NF kappa B signaling pathway. It's predominant in almost all types of cancers. The NF kappa B pathway is subdivided into two pathways: canonical, sometimes called classical, the old one, and non-canonical, now called the alternative, the new one, the new side of the pathway. The canonical side of the pathway has a rate-limiting enzyme called IKK beta, inhibitory kappa B kinase beta, and it's responsible about the activation of a biomarker called P65. Sometimes they call it REL-A. Okay? The non-canonical side, on the other hand, the rate-limiting enzyme is IKK alpha, and is responsible about the phosphorylation and activation of what we call P100. So it phosphorylates P100 to form an active P52 that enters to the nucleus and do all the transcriptions required. Now, the coming slides will focus on the non-canonical side of the NF-kappa B pathway. We will focus on IKK alpha and P100 phosphorylation. So you will hear these words coming in the coming slides, frequently in the coming slides. Simply, the canonical side of the pathway, the old pathway, has a rate limiting step of IKK beta, but IKK beta, it is the predominant enzyme, but it lives in combination with other IKKs. One of them is IKK alpha as well. But the predominant is IKK beta. And it's responsible about releasing IKB alpha and then allowing P65 to enter to the nucleus and then do some transcriptions to form proteins needed for the cancer cell. Now, if we manage to inhibit IKK beta, then we will stop the formation of P65, we will stop P65 going into the nucleus, i.e., we will starve cancer cells from important proteins, and then supposedly the cancer will die. On the other side is the non-canonical pathway. So upon activation of this pathway, IKK alpha, which is the predominant enzyme, will phosphorylate P100 into active P52, and then P52 goes inside the nucleus and does almost the same as P65. So if we block these enzymes, we're probably gonna inhibit the entire pathway and kill cancer cells. We tried to use IKK beta inhibitors, but we found that IKK beta is actually important for cell survival, normal cell survival. So by inhibiting IKK beta, we produce a lot of side effects and less efficacy. So we've shifted to IKK alpha, to the non-canonical side. And by inhibiting IKK alpha, we managed to find that there's very specific effect on cancer cells and no effect, almost no effect on the normal cells. So we carried focused our all science on IKK alpha. We still use IKK beta as a reference though, for selectivity. See that those pathways that I show you were very simple. They are a little bit complicated. They look like this, even more complex than this one, but I'll stick to the simple one. Let's see some results. So starting off our experiments, we would like to try to stimulate the pathway in our cells. So I use some GBM cells called T98G cells, and we try to stimulate them using an agonist. So the agonist right? Okay? No. Okay? I might need some help with this. Sorry. No, it's not working. Sorry again. Okay. This is making trouble. All right, let's continue. So I started to stimulate the pathway using an agonist, and that agonist is called tweak. So it's a TNF weak inducer of apoptosis. And the techniques I used in my experiments called Western blotting. So Western blotting helps me to identify the amount of the proteins of target inside the cell. And the results of the proteins of Western blotting experiments comes in these bands. The more intense the band is, means that there's more protein within the sample. So I used a tweak to try to increase the phosphorylation of P100 there. So the phosphorylated P100 is supposed to increase by using tweak as an agonist. So I can see that by increasing the concentration of tweak, the intensity of the bands increases. And I can measure these bands' intensity and make them in numbers and then form this kind of bar chart. So the bar chart will help a lot. So you can see that there is an increase in the concentration of P100, phosphorylated P100, by increasing the concentration of the tweak. Most of the literature says that we best use 100 nanogram per ml of tweak. Only one study was showing that 10 nanogram per ml was enough. And this study, my experiment here, showed me that yes, 10 nanogram per ml is actually very equivalent to 100 nanogram per ml. So I don't need to use not 100 nanogram per ml. So in the rest of my experiments, I use 10 nanogram per ml of tweak as an agonist. So now I found out that the NF-kappa B, the non-canonical site, is active in these cells, and now I need to investigate more about the tweak before I start to use the inhibitors of the signaling pathway. Next comes the effect of tweak again on the phosphorylated P100, but this time I was concerned about time, not concentration. Now I know that I'm going to use 10 nanogram per ml, but what does it do over time? If we use the same concentration over a 24-hour period, what happens to the phosphorylation of P100? And I managed to see, starting from 30 minutes, 1 hour, 2 hour, 4 hour, 8 hour, 24 hours, that after 8 hours, there was a decrease. There was a nice steadily increase in phosphorylation of P100, but after 8 hours there was a dip. The presumption was that there was a complete depletion of the IKK alpha enzyme, and then the cells need to regenerate and resynthesize IKK again. Why there's a big gap between 8 and 24? Why don't use 16 hours maybe, or 12 hours maybe? Why there's a gap? Does anyone know? Because between 8 hours and 24 hours, I was sleeping at home. It was after midnight already. I cannot do my experiments after midnight. There was a policy in the university that you cannot use the lab after midnight. So I couldn't utilize the lab after midnight. So there's a bit of gap. I am still concerned about what happens after 8 and 24 hours until today. Right. So we know that now P100 is phosphorylated by tweak. Now we want to see if the phosphorylated P100 will have an effect on the formation of P52. So we managed to examine P52. So I'm expecting that by phosphorylation P100, the P100 itself will decrease by time, and the formation of P52 will increase by time. So again, if we see the bands, P100, yes, it decreases. That's because of the phosphorylation. And P52 is actually increasing by time. And the figure actually shows that. There was a significant effect. Good. How about the amount of P52 inside the nucleus? It is a bit of a different preparation for the experiments. We first use a cytoplasmic sample. Now we need to use a nuclear extract. So I extracted the nucleus from the sample and tried to see the P52 inside the nucleus. And again, we can see that the P52 is actually increasing inside the nucleus. Once it's inside the nucleus, there will be a P52 DNA interaction to form different kind of proteins. Good. So now we have the proof that tweak can activate the non-canonical side of the pathway. Now, is it selective to the non-canonical side, or does it work also on the canonical side as well? So to prove that, I was keen to try it. Let me try the tweak on some of the biomarkers of the canonical side and see what happens. So I tried it on IK beta. So I'm expecting by stimulating IKK beta, IK beta will decrease in number and release P65 that goes inside the nucleus. That was my expectation. So upon stimulation with the tweak by using 10 nanogram per ml, tweak was able to not stimulate the canonical side of the pathway. So that means that tweak is selective to the non-canonical side and makes me happy. This actually haven't been proved by any study before. This was the first study to prove that the tweak is actually selective to the non-canonical side, but not the canonical. In reasonable concentration. In higher concentration, it loses selectivity. Good. Now, our chemistry department in Strathclyde University thankfully gave us some compounds. After a long time of experimentation, Dr. Ishaq said it takes about 10 to 15 years to come up with one compound to be released in the market. Actually, these guys have been working on some compounds for 25 years now. And finally, thankfully, these two compounds, the SU1433 and the SU1644, which are selective IKK alpha inhibitors, small molecules, have been patented and taken by Johnson & Johnson. They purchased them. They are now going into further experiments. Now they are going in vivo experiments, and hopefully in the future we will have some also patient trials. So these drugs might see the future light, hopefully. So I use them. If you see that SU stands for Strathclyde University, and then 1433 is the number of this molecule, 1433. So they reached to 1433 remodeling of compounds to reach to this one. And then 1644. This is the molecule number 1644. And actually they actually reached more than 2,000 compounds. They didn't stop here. They continued remodeling and trying to modify the drugs, the compounds, but they found that these two are the best. So they continue with these two now. So I use them as selective inhibitors of IKK alpha. Again, back to the phosphorylation of P100. Using now the compound. So I stimulated the pathway using tweak. And after the stimulation, I used the inhibitors to see what happens to the phosphorylation. When we used tweak, we saw that the phosphorylated P100 actually increased. Now when we use the compound, the inhibitor, it actually decreased. So the concentration of phosphorylated P100 decreased. That's a good news. How about the formation of P52? Does it increase? No, it actually also decreases by using increasing concentration of SU1433. So that means I'm expecting that P52, as long as it decreases here, it will decrease inside the nucleus as well. So we are depriving the nucleus, we are depriving the cells from having these proteins to help it to characterize into cancer cell. So inside the nucleus, yes, P52 was also decreasing inside the nucleus. That's nice. That was 1433. How about our friend 1644? On phospho-P100, it actually also shows decrease. With actually an IC50, an IC50 measures the potency of the drug, how potent the drug is, simply. So showing 10 times more potent than 1433. So 1644 is actually 10 times more potent than 1433. I'm even using less concentration. The 1433 I reached 10 nanogram. With 1644 I reached 3 nanogram. When I used 10 nanograms, actually the normal cells died. I don't want the normal cells to die. And then the effect on P52 formation, also good, not bad. And then the translocation of P52 inside the nucleus will also decreasing. Now let's see the effect. We managed to find that these drugs inhibit the cellular effect of IKK alpha. How about the cell viability and the clonogenicity of these cells? Does it decrease by using these compounds or not? Is it affected? The phenotypic characteristics of the cancer cells, is it affected? So this is a cell's viability study using MTT assay, and we can see that using 1433 there's a decrease in cell viability. Also 1644 that shows decrease in cell viability. And as a reference, I used something called ML120B, an experimental compound, which is selective to IKK beta only, not IKK alpha. Also showed a cell viability decrease. Why is that? Because IKK beta also can kill cancer cells, but it also kills normal cells. This is a clonogenic assay to see the formation of colonies over time. So these are the cells here, the colonies showed here. And by increasing the concentration of our SU compound, you can see that the number of colonies actually decreases over time. So it decreases the colonization of cancer cells. 1644 also decreases the number of colonies over time. It does look bright future for these compounds. Now, in the UK, my supervisor told me that they actually shifted to animal studies on these compounds, and hopefully in the future we might see them in the market. So, my take-home message is simply that the future of managing cancer looks really bright. Thank you very much again.
Thank you so much, sir, for this wonderful session.
I hope it was interesting. I hope it was an interesting talk. Was it? Wait for a question, yeah?
Now the session is open for question and answer. If anybody want to ask question, please raise your hand. So the first question I want to ask you, sir. I'm also from Ecology department, sir.
Oh, okay, good.
So, according to my basic knowledge, the neurons can't divide. So, in cancer, we say that cancer is uncontrollable division of cells. So, what is the difference between brain cancer and normal cell cancer?
Nice question. Nice question. Brain cells or brain cancer cell division is fast enough that it looks more like bacterial cell division. And that's one of the most challenging things with the brain cancer cells. I'll give you a hint. When I was doing my experiments, I have to grow these brain cancer cells in the lab. And I have other colleagues who works on other different types of cancers. So one of my colleagues works also on prostate cancer, and others on breast cancer, and I was doing brain cancer, okay? So, my colleague with the prostate cancers, he needs to grow the cells in the dish and then keep feeding them for some time until they grow in the dish, and then use them and utilize them, okay? So it takes him about 7 to 10 days for the dish to be full of cells so he can use them. We call them confluency. 80% confluence. Brain cancer cells. So 7 to 10 days, okay? Imagine. My brain cancer cells. I grow them 9 o'clock in the morning. The dish is full by the evening. 4 o'clock in the evening. It's full. Or if I don't reach them by, like, say 5 o'clock, it will be more than 80% confluency. I cannot do my experiments. It's that aggressive in cell division compared to other cells. It's so hard to deal with them. And it's even frightening actually to see that rate of division in cancer cells, in the brain cancer cells mainly. Did I answer your question?
Yes, sir.
And this is one of the challenges actually in managing brain cancer. So what we, as I said, they debulk the cancer by surgery, but they cannot take the entire cancer tumor because they have to leave some normal cells. They don't want to affect and damage the normal cells of the brain. Otherwise, it's going to cause a disaster to the patient. So they leave some of the cancer cells available, and then what they tend to do, they start to use chemotherapy and radiation therapy to try to remove the remaining cells, percentage of cells. However, because it is so fast in cell division, it regrow again. And sometimes you need to do the surgery again after a few months. That's one of the challenges of managing brain cancer.
Thank you, sir. And the second question is, in case of brain cancer, the main hurdle is blood-brain barrier. So if we want to achieve targeted drug therapy, so how we can do that?
Very nice questions. You should work with me.
Thank you, sir.
This actually was our challenging question when we were in the UK. Now we formulated these compounds, but we need to also think about the pharmaceutical formulation for these compounds. And it was so hard, as she said, for the drug to pass the blood-brain barrier and act on the cancer cells. Not only that, but also cancer cells start to make another barrier on itself, making it even harder for the drug to reach to the cancer cells. So one of the ideas that came in our mind, I'm not the one who's dealing with the pharmaceutics, but there are guys doing the pharmaceutics, right? We discuss these things. So they came up with the only solution of injecting the drug directly intracranial to the cancer cells. That was the only solution. According to the characteristics of these compounds, the only way is to directly inject them into the brain. Yesterday while I was seeing these posters, one of the posters was about glioblastoma. And they were thinking, I can't remember one of the girls there, she was thinking of developing a nasal gel for the drug delivery system for the brain. That was a nice idea, very nice idea. However, I believe that this would cause a lot of side effects, one of which locally in the cells of the nose by itself. Necrosis of the cells of the nose, because most of our anti-cancer drugs are cytotoxics. But it's a nice idea, very nice idea.
Thank you so much, sir.
Right.
Next, anyone having any questions, please raise your hands. Anybody from students want to ask question, please raise your hands.
Yeah. It's either you understood the entire presentation or you didn't understand anything. I hope that you understood my presentation. That's why you don't have any questions.
Good morning, sir.
Good morning.
In some of your slides, you mentioned the Western blot image, right?
Yes.
So somewhere you mentioned glyceraldehyde phosphate as a housekeeping protein, and somewhere you mentioned nucleolin. So this housekeeping protein is dependent on the amount of protein you have loaded or else increase.
Yeah. It actually depends on the amount of protein inside the cells. So the cytoplasmic protein. So what we tend to do, do you work on Western blotting?
Yeah, I'm from pharmacology.
Nice. That's very specific questions about the Western blotting. So we use the loading protein at the end. So it could be, as you mentioned, nucleolin if we use nuclear extracts, or GAP-DH if we use cytoplasmic extracts. So the loading protein, the GAP-DH, the loading antibody there, is only as a reference of the amount, or let's say the integrity of the amount of proteins in each sample. It just represents that each sample has the same amount of protein, approximately the same amount of protein. Did I answer it?
Yes. Actually, I'm going to do Western blot, so.
Okay. It is very challenging, it's tough, but it's one very beautiful experience. It will take you days to finish, but it's nice. Usually when we do Western blotting, it sounds simple, but the thing is we have to first grow the cells and then try to treat the cells and then make the samples This only takes about a week, and then we, in my case, of course, my friend in prostate cancer requires 2 weeks for this process. And then I do the Western blotting. The Western blotting, the one I was doing was denaturing, so it takes about 2 days. So the first day of Western blotting, we try to add the proteins and then try to put all the required apparatus there. The second day is the exploration. So we want to find out our results, the film that you've seen in one of the slides. And it is so exciting when we are in the dark room waiting for the results to see, oh, did I get the results right or not? Because sometimes you get blank results and you start all over again from the beginning, like a 10 days work starts again. But yeah, it is very nice to see your results, always nice. It's like, and the cells, by the way, if anyone's dealing with cells here, cell lines, I used to treat them like my sons and daughters. So I go to them and speak to them early in the morning. Hey, good morning, how are you today? Are you okay? Everything's fine? I think they communicate with me somehow. We don't want, it's tough to work with also cells because you don't want to lose the cells at any certain point. One single contamination is a disaster because it means you have to take all the cells, throw them away, and then clean up all the entire lab again, and then seed new cells and regrow them again. It's a hectic process. So you have to be very good with the aseptic techniques. Yes?
Hi, good morning, nice presentation.
Thank you very much.
Just I would like to ask a simple question maybe for you. Some people will say anti-cancer activity, some people will quote like a cytotoxic activity. What is the difference between those two?
Good, very good. Cytotoxic, the question what's the difference between anti-cancer and cytotoxic? Anti-cancer is quite broad term, but cytotoxic means that this drug will kill normal cells and cancer cells. But when we say anti-cancer drugs, we might talk about all the anti-cancer that I've mentioned in my presentation. Targeted drug therapy, immunotherapy, for example, even cytotoxic chemotherapy, all are covered by the name of anti-cancer drug therapy. Right? This actually applies to antibiotics the same way. So we have antibacterial, antiviral, antifungal, but the broad name is antibiotic.
Anybody having question?
I always tell my students, don't be shy to ask questions. I don't have a stick to go on. Why do you ask this question? Don't ask it. No. Ask the questions, be happy. If I don't know the answer, I will say fairly I don't know the answer. But I try to find out.
Good morning, sir. So even I am working on NF-κB pathway.
Nice.
With psoriasis. So I am working with two pathways, MAPK and NF-κB, and they are cross-talking. Like they have some interactions in between.
Can you repeat the two pathways again?
MAPK and NF-κB.
MAP kinase and?
NF-κB.
NF-κB.
Yeah.
Which part of the NF-κB?
P65 for NF-κB and P38 for MAPK.
Okay.
So they cross-talk.
Yes.
Okay. So are there any other, like for example, for me was NAPK, is there any other pathways which you can target like this? So like, stopping it from the start only, not going through NF-κB, because the MAPK actually, it forms a cascade of inflammation.
Yes.
So is there any other pathways which will stop it at the start only, not going to NF-κB?
So do you have any inhibitors that you use?
Yes, I have my drug which I'm using. Not.
Is it a small molecule?
I wanted to use a small molecule, but sir, it's not feasible.
That's the point. I want to get to this point. Well, she's working with some of the signaling pathways as well. But the thing is, if we have small molecules, it is beneficial to go inside this signaling pathway and try to find very specific rate-limiting step. If we don't have small molecules, it means that the drug cannot actually go inside the cell. So we might work somewhere outside the cell, a little broader. It's a good idea, but if we block broadly, I'm expecting a lot of side effects as well. To avoid these side effects, that's why scientists came up with the small molecules, trying to get inside the cell, very specific, and that's why it's called targeted therapy. To try to avoid the side effects. but it's very good. Now, other pathways. I cannot tell you what other pathways you can use because she mentioned NF-kappa B, yet she's working with psoriasis. When we started to work with NF-kappa B, we started as anti-inflammatory drugs. Our aim was anti-inflammation. But we saw potential with blocking this pathway to produce anti-cancer effect. So we moved to anti-cancer drugs. And inflammation was taken by someone else. So NF-kappa B and all these cytokinic pathways have both effects as anti-inflammatory as well as anti-cancer. But I cannot specify something for you like any other pathways because, as you said, they are interconnected. You have to do the network analysis. You have to analyze whether this is important to block and will it yield effect or side effects. Otherwise, I will go for, I'll do ERK, I will do MAP kinase, I will inhibit IKK, I will inhibit whatever I want. But I would like to be very specific. Okay? And it differs between disease and another, inflammation and cancer. It differs from cell to cell, which is the predominant NF-kappa, which is the predominant signaling pathway in this kind of cells, which is the predominant pathway in this kind of disease. That's it. Thank you for your question. I've got really nice questions today. I'm lucky.
Good morning, sir.
Good morning.
Thank you for your excellent session, sir.
Thank you.
Here's a small question for you, sir. Usually, chemotherapy in cancer have lots of side effects.
Yes.
So, are there any future chances to decrease these side effects, especially targeted to chemotherapy?
Yes, yes. That's why we have this future of anti-cancer drugs. Immunotherapy is meant to kill cancer cells specifically and has fewer side effects. Actually, most of the immunotherapeutic drugs nowadays, like the monoclonal antibodies, they have beautiful effects, yet very less side effects. The most prominent side effect with the monoclonal antibodies is hypersensitivity reaction. That's the most prominent. But there could be some other side effects, like immunosuppression maybe or something like this. But they still have less side effects than the older fashioned cytotoxic drugs. Yeah? The future is really brighter. Thank you very much.
And sir, hello, is there any futuristic approach to avoid side effects of cytotoxic or anti-cancer drug?
The formulation guys are working on it. They are doing some liposomal formulation, some nanoparticles and stuff like this to try to come up with solutions. One of which was, for example, I've seen some of our colleagues using what they call caging. They cage the cytotoxic drug with some sort of polymers, and then they give it to the patient. It will reach the site of action, and then it will release the cytotoxic drug within the cancer cell. That's one way of reducing the side effects of the older cytotoxic drugs. Yeah, I've seen so many research actually in this part. The good thing is, I have lots of questions. That means there are so many people now interested about inflammation and cancer, and that's good because they are, especially cancer, is the disease of the era, of the century, and we have to work on it hard.
Okay, thank you so much, sir.
Good morning, sir.
Good morning.
Your lecture is very knowledgeable, sir. Sir, my question is, brain tumor is curable after surgery or chemotherapy, but some malignant tumor are not curable. Any sir alternative therapy for this malignant tumor after surgery or after chemotherapy also not treatable, especially malignant tumor, sir. But this breast cancer or ovarian cancer, any alternative therapy for?
Okay. I think I got your question. First off, brain cancer is still not curable. Yes, we can do debulking surgery. We can use radiation therapy. We can use some chemotherapy that we have. It's only temozolomide and no bevacizumab. But still, it's recurrent. It's recurring again, and the mean survival period is only 15 months. And the survival period is actually terrifying, is miserable. So still brain cancer is not curable. But hopefully we can come up with some cure for it in the future. Or maybe increase the survival period with better quality of life. Other types of cancers, there are a lot of research going on other types of cancers. Most of the cancers nowadays, thankfully, are curable. Yes, they make a lot of side effects, or the drugs make a lot of side effects, but at the end the patient will be relieved from the cancer. At least he will live fine for a very long time. One of which is my father. He passed away a few years ago during Corona, but he was fighting prostate cancer for like 10 years. Thankfully, he managed to be cured from prostate cancer by surgery and some chemotherapeutic agents and radiation, and he lived for 10 years happily. So there are types of cancer that are curable, but we're still struggling with some other types of cancer that we need to find a cure for them. Did I answer your question? Thank you very much.
So, it's me. So when you said reoccurrence of cancer, so you also mentioned about cage, which is like a 3D printer cage, the
Yes.
drug inside the 3D.
Yes.
So can we do, as in, give the cage, like the 3D printer liposome inside, keep the drug in the body, and whenever the cells divide and the body detects some signaling, the dividing of the cells, then only it releases the drug, so that there is no reoccurrence only of cancer. Can this be possible?
I don't have the answer for this question, unfortunately. I haven't worked on pharmaceutical formulation when I was doing I was mainly molecular basis. I haven't seen any trials on these kind of compounds and the long-term efficacy of caging, for example, these kind of compounds. I haven't really read about it, I haven't seen it, I cannot give you an answer for it. But it is interesting to read about it. I will read about it. You can read about it. Everyone else can read about it. Sorry about this. Thank you very much. Nice question though. She's asking really good questions.
Anybody is having question, please raise your hands.
A student?
Hello, sir.
Hi.
Thank you for your excellent session.
Thank you very much.
As targeted cell therapy can develop resistance. Cell can develop resistance of our prepared drug. So what can we do? Because cancer cells can have mutations.
Yes. Very nice question. Another beautiful, very beautiful question. Yes, we have resistance developing with some types of cancers. One of which is breast cancer. It develops resistance, and it's very hard when it develops resistance. What we tend to do in the clinic, we try to find out we go outside the guideline and we try to use other types of therapies available to target these resistant types of cancer. However, not all cancers can have resistance. Some of them, yes. And when they become resistant, it's very hard to tackle this resistant species or this resistant type of cancer. And the death rate is higher than the other types of cancer.
Sir, as cell divides more faster in cancer.
Sorry, again?
Cell divides more faster in cancer.
Cell division?
Is more faster in cancer.
Yes.
So DNA's changes are also more faster.
Yes.
So mutations are the one who are resisting us from cancer treatment, perfect cancer treatment. So can we have a drug that can change according to the mutation or in future?
You're very well understanding cancer, by the way. There are some slides, there are some things I've done and techniques and experiments I've done in my project that I didn't share today because it's quite complicated. She was talking about gene mutations. Yes, there are gene mutations. And one of which was, in my brain cancer, for example, we found that there is a gene mutation called human telomerase transcriptase enzyme, or gene. It's overexpressed in most types of cancer. And there are two subunits. About 60% of all types of cancer has this gene mutation. And there are two subunits of this HTR gene. The C250T, which is more predominant, about 80% of the 60% of HTRs are C250T mutations. And there's C228T mutations. And I was very concerned about this. So I've done other experiments called EMSA assay. I was thinking about finding out the protein-DNA interaction and the effect of inhibiting this signaling pathway on the gene mutation, HTR specifically. Another gene mutation in brain cancer is the MGMT. Very well known. Any patient with brain cancer will do the MGMT test. If he is positive, he will go under different kind of guideline of therapy. If he's negative, he will do another guideline of therapy. But nobody actually thought about HTR. It is more predominant, specifically in brain cancer, than the MGMT. So I proved actually that HTR is important, and these agents that I'm using are actually specific in inhibition to the C250T mutation of the HTR. So I also patented another thing, a new way of diagnosing brain cancer, not only the MGMT positive, but also HTR positive, C250 mainly, and then I patented two molecules here, the SU1433 and 1644, to be selective inhibitors of brain cancer C250T mutation, HTR. Thank you very much for this question. I raised this point just for you.
Thank you so much, sir.
Thank you very much. That was a very specific question, very nice. So yes, as you mentioned, we can bring drugs that are specific for certain gene mutations in cancer.
Hello, sir. Sir, thank you for this session.
Thank you.
Sir, very simple question for you. How the drug identify or differentiate the healthy cell and cancer cell?
Can you repeat the question?
How the drug identify differentiate cancer cell and healthy cells?
Cancer cell and?
Healthy cells.
Healthy cells. How the drug can identify the difference between normal cells and cancer cells. Cytotoxic drugs cannot identify this. But our molecules here can identify it. How? Because of the overexpression of NF-kappa B pathway in the cancer cell only. So the normal cells will not overexpress this pathway, it will be normal. But in cancer cells, this pathway will be overexpressed. That's why our compound will work only on the overexpressed pathway. So specific to the cancer cell.
Thank you.
Nice questions. Very nice questions, Wallah.
Very nice presentation, Dr. Haider. We really enjoyed it.
Thank you.
Just a very, maybe because I'm trying to be very optimistic about cancer, since also I have one of my relatives actually passed away because of cancer.
Sorry to hear that.
It's okay. The only thing that I'm worried about is the time that it takes one drug to develop and to come to the market. So do you think that they will try, since this is really very promising, do you think they will try to accelerate? Yes, actually you have even anticipated what would be my question. So are they planning to really accelerate like all the trials? You said they have started already the animals at the moment. Are they planning to accelerate it and start even the clinical trials?
In terms of accelerating the trials or the studies on specifically talking new era of medications like targeted drug therapy is very hard. Because we still not fully understanding how these drugs affect is, and we can predict the side effect, but actually we cannot see the side effect. We have to do some molecular basis, some animal experiments. We have to be very patient with it to see all the profile and before we release the drug to the
I
market.
can help you predicting, by the way, using BD.
Yes, exactly. Why not? Yeah, we use different kind of models, by the way, the computed models. it assists and helps in reducing the time as much as we can. But yes, unfortunately, it's not really easy to accelerate the studies on such molecules. It will need some time. I probably assume from today, maybe 6 years, if they really work hard, in 6 years, these drugs might really get approved and released in the market.
Hopefully.
Yeah.
Thank you very much. Thank you.
Very much. Thank you very much.
Thank you so much, sir. I think our question and answer session is over. Give him a big round of applause for this wonderful session.
I would like to say something. This is my first visit to India, and I'm pretty sure it will never be the last. I love you so much, and I will come back again. I'll do this again for you. Thank you very much.
Thank you so much, sir, for this compliment. Your session was very informative and you have cleared our doubts. So, on behalf of our organizing committee, I would like to say very much thank you so much, sir, for having this informative session. Thank you, sir.
And unfortunately, because we need to run for the flight, we will not be able to continue this session with you today. So we need to leave to get to the airport and then fly back home. But I promise I'll come back again.
Thank you so much, sir. Now, I request respected Mrs. Mazur ma'am and respected Dr. Vishal Babu sir to felicitate our another speaker, Dr. K. Suresh Babu sir. Thank you so much, sir. Now I request Joe ma'am and Babar sir to felicitate our fifth eminent resource person, respected Dr. N. Shankaraya sir. Thank you so much, sir. I hope our participants are gaining lots of knowledge from this international conference, and keeping this, we are moving ahead to our fourth session of this international conference. I know all the first three lectures are very informative and knowledgeable. So, for the next session is of Dr. Suresh K. Babu sir. To introduce Suresh Babu sir, I request Alisha ma'am to give his introduction.
I, Alisha Sheikh. Good morning, everyone. It's an absolute honor to welcome you all to the International Conference on Advances in Pharmaceutical Science Research, Traditional to Novel Approaches. Today's session promises to be both enlightening and inspiring as we delve into cutting-edge advancements in natural products. How have they contributed to health and society? We were privileged to have with us a distinguished scholar whose pioneering research in drug discovery, natural products, and medicinal chemistry has impacted countless lives. It's my pleasure to introduce Dr. K. Suresh Babu sir. Currently, he is serving as a senior principal scientist in the Department of Natural Products and Medicinal Chemistry at CSIR, Indian Institute of Chemical Technology in Hyderabad. He earned his BSc and MSc from Kakatiya University and completed his PhD at CSIR IICT in the year of 2005. He later pursued postdoctoral research at the University of Mississippi, USA from 2005 to 2007. While coming to his professional experience, Dr. Babu sir has held various positions at CSIR IICT, progressing from scientist fellow to senior principal scientist since 2019. His research area includes on drug discovery, natural products, and medicinal chemistry. While moving to his publications and patents, Sir published more than 200 research papers, 2 reviews, and holds 14 patents with 4,500 citations and has an H index of 33. His work has led to the licensing of 2 patents to MS Sami Labs, Bangalore. 2 processes technologies, development of process for extruded Nutra-Food products for management of metabolic disorder, anemia, and malnutrition. These products are now available in the market. Dr. Babu sir has received multiple awards, including the OPPI Young Scientist Award and Andhra Pradesh Academy of Sciences Young Scientist Award for the year of 2010. He received Dr. A.K. Singh Memorial Young Scientist Award for the year of 2019. He received Best Performance Award for the year 2011, conferred by Director of IICT. He has been elected as a fellow of various academies, including the Telangana Academy of Sciences in 2020, and also received the Senior Scientist Award from ABAP in 2023. As a mentor, he has guided 30 PhD students to completion with 5 ongoing. He also serves as a scientific consultant and an editorial board member for journals in pharmacognosy and chemistry. His contributions to pharmaceutical research has significantly impacted both academia and industry. Now I would like to hand over the session to Dr. K. Suresh Babu sir, who will take us to deeper into natural products. How have they contributed to health and society? Please welcome Dr. K. Suresh Babu sir with a round of applause to share their insights on this topic.
One second. Sorry to interrupt you, sir.
Right. Fine.
So our guests from Oman are leaving, so we are giving a bit of goodbye to them from our organizing committee and Dattakala Shikshan Sanstha. So I request Jokha ma'am, Heather sir and Ishaq sir to give some feedback regarding this event and cultural events we had.
Honestly, I am very overwhelmed again by the hospitality, by the organizing committee, mashallah. It has been a pleasure to be in India for the first time, and even more pleasure actually to see how hospitable you are actually. Respectful, whatever Haider mentioned actually, we were discussing that, how respectful the students, the staff, everybody here. To be honest, I just started to think that I need to visit India more. The diversity, the culture event have given me a huge memory actually that I will never forget, and I will take it, I think, with me now going back home. Seeing the culture starting from the Himalaya, and please correct me if I'm wrong, until the south, I think near the beach. Honestly, amazing. Amazing customs, dancing, music, very energetic, and the food, wow. Despite being spicy, still. And the really thing about India, despite the diversity that we have seen it in the past few days, still really, you're really one nation. We cannot see that diversity. Only we saw it when we had the cultural event. So really, this is the essence of India basically. So thank you very much. Thank you.
Thank you so much, ma'am. This will be the compliment for us.
Thank you very much. I cannot say anything enough. I mean, the hospitality. I felt yesterday that I'm a superhero. Lots of pictures and photos here and there, and then when we received lots of things beautifully. I haven't seen anything like this before in my life. So thank you very much. I learned this yesterday. Thank you very much. And for the students, all the best in your career and hopefully in your future as well. You'll be the best pharmacist ever. Thank you very much again.
Thank you so much for having us. I really thank the management and the faculty members and the organizers for having us. Literally, you have proved our Indian customs, that is Atiti Devo Bhava. Thank you so much, and really proud of an Indian and really proud of an Indian Muslim. Thank you so much. Thank you all.
Thank you so much, sir and ma'am. I request Suresh sir to continue his session.
Yeah. Good morning, dear students. I would like to thank the management and the organizing committee for having me in this international conference. particularly for giving the opportunity to share my views regarding the natural products and how they have contributed to the health and society and for well-being of human being. So I just take you to the essence of natural products and what are the drugs derived from different sources of natural products like plants, marine and algae and so many, and how they have been discovered and what are the approaches for the natural product drug discovery. And I will also touch upon some of the quality control aspects of herbal drugs where in the current scenario we are facing that quality control problems, otherwise natural products are very good drugs. So before going, just if you think of we have the different distinct seasons in Indian climate, but because of several reasons there is a lot of changes in the atmosphere or environment, and that there are many causes. Like best one is pollution, and we are also removing the trees, and because of this we are facing a lot of problems. There is no rains in the rain season, and temperature now in Hyderabad the temperature is almost 38 to 39. In some other places it's already summer began, 39 class 39, but it's not a real summer, February as per this one, but because of all man-made things. And another thing is the plastic. When plastic was discovered in 20 years back, we were so happy that will make our life very easy. But now we are facing a lot of problem to destroy the plastic. And even a recent study, it was microplastics also traced in the even mother milk, which so far we claim that is the purest thing in the nature. And several other even traces also found in our blood also, where it has come because of the plastic usage, and we are taking even tea in plastic cups, paper cups, where there is a wax kind of polythene coating, and we are also using the Teflon coated pans. So after 1 month or 2 months, the Teflon coating will be gone. So where are all the Teflon went into our intestine. So there are, if you can say, there are several hundred examples. So that is a major reason for the cancer. I think maybe another 3-4 years or another 10 years, I think 1 in 10 people will be detected as a cancer. So nowadays technology is very high, and the detection rate is also very high, and the prevalence of cancer is also very high. So if you see all these incidences, there is a challenge for drugs for different diseases, global health care. Definitely, because there are several reasons, as I mentioned you, non-tropical deforestation, increasing fossil fuel, which leads to the global warming. And because of all these environmental changes, there is a new incidence of new global diseases like SARS, avian flu, and also increasing the drug resistance is also one of the biggest problem. We must have taken a lot of azithromycin, dose 650, Montac-LC during our COVID 1 and 2 period. So now azithromycin 5 doses is not enough because already immunity is developed in our body. So that is also one of the biggest reason. And because of all these instances, there is a new instance of new diseases also. Now it is GBS syndrome. I think you must have heard it. GBS syndrome now running other than the virus. So these are the major diseases. AIDS, tuberculosis, diarrheal diseases, malaria, hepatitis B, SARS, avian flu, and tobacco-related diseases. You can see every year more than 1 million deaths are happening, and more than almost 500,000 cases of new cases are finding. If you can think of the treatment, there is no globally available treatments for the diseases in the particular the critical stage, and of course modern lifestyle is also one of the biggest problem for our healthcare. And nowadays, all complete modern lifestyle. See here, the whole family is enjoying with cell phones in the one sofa and watching the TV, almost to home theatre culture. Earlier, our forefathers used to sit in the dining table, or once all family used to take dinner or lunch, but now that culture has gone. So, and this guy is busy with taking the burger while working in the computer. So what happens is in that case, we don't have any sense that our stomach is full. We eat more food, and that is one of the major cause of diabetes and obesity, which is a major problem including adults and nowadays even children also. So this also global healthcare is definitely in crisis, and getting a drug is more expensive. Almost it costs about $1 billion, and there is no drugs in critical global diseases. We have seen in COVID 1 and 2 period, even if you are affordable to take medicine also, there is no drug for COVID virus like other viruses. And in this junction, can we think of our traditional medicines like plant-based medicines or Ayush-based medicines, so which are major difficulties, poor quality control, but in this present scenario of the revolutionary techniques are evolved in the analytical and biological techniques. So in this scenario, we can come across all the quality control parameters of herbal drugs. So this traditional medicine, what is a traditional medicine? Because in those ancient times, people with their limited knowledge, because there was no education for our ancestors, based on their knowledge, they have identified plants around them, surrounded by them are like medicinal plants or food plants and poisonous plants. So the categorization and the knowledge has been rooted to the generations together, formed as a traditional medicine, which we call upon like Ayurveda, Siddha, Unani and homeopathy. Every country has its own system of medicine, like Egyptian medicine, Chinese medicine and South African system of medicine. All are formed like this only. When coming to India, traditional medicine can be classified into two categories. One is codified system, another one is non-codified system. Codified system comes under the Ayush ministry like Ayurveda, Siddha, Unani and homeopathy, and non-codified system, folklore medical system, where majority of the tribal people till date they are using this folklore medical system, but there is no scientific basis, but they are doing much better. Of course, these two systems have contributed equally to the primary health care in India. So even developed countries also, the traditional medicine is in practice, but not the same name. This is called a complementary medicine, alternative medicine, non-conventional medicine, but the raw material is same medicinal plants. And of course in US that is called botanical medicine, in Europe that is called nutraceuticals, in India it is called traditional medicine or Ayurveda medicine. Like, see, in developed countries like here pictures, the traditional medicines also available over to the counters like Iran, Colombia. These are some of the pictures of over to the counters in developed countries. And this kind of people you can come across when you visit the tribal people or interior areas where there is no accessibility of doctor and hospital. These people are called traditional healers or folklore medicinal healers. And recently, Government of India also conducted a survey regarding the existence of these people. Almost 4 lakhs to 5 lakhs traditional healers are existing in the entire country, north through south, east to western. So these people are serving the surrounded people, surrounded by the nearest villages and the neighboring people by giving the folklore medicine. And these people got the knowledge from their fathers and forefathers, and but they are doing much better. They are doing good support to the neighboring people. And majority of the folklore medicine was practiced by the tribal people, where India is the largest tribes, and scattered in the different regions like Himalayan region, northeast region, and south region, western region, Andaman Nicobar Islands. Even in some part of India like islands, Andaman Nicobar Islands, people still they are using completely plant-based medicine. There is no access of even paracetamol, single, even antibiotics also. But they are not doing very good when compared to the modern civilization people. And based on this limited information, recently World Health Organization has conducted a survey regarding the utilization of the herbal medicine, and it really revealed that almost 80% of the world population relies on the plant-based traditional medicine for their health care and primary health care needs. And further, it also listed another 21,000 plants which have been reported very highly medicinal uses around the world. And when coming to India, India is known about the botanical garden of the world. And we have very distinct 60 hydroclimatic zones and flooded with 45,000 different plant species, and majority of the species were used in our traditional system of medicine like Ayurveda, Siddha and Yunani, and further it is also listed another 5,000 products which are included in the pharmacy of Ayurvedic treatments. And these are the major natural sources like plant kingdom, microbial world, animal sources, marine world and the innocent toxin. Among the 5 sources, plant kingdom has surveyed a lot in the form of Ayurveda and other drugs. And microbial world is also one of the good sources where the best example is penicillin, where penicillin was discovered from microbial world. And animal sources also some of the drugs were discovered which are in the present market. Best examples are epibathalins, which are discovered from the skin, flax skin, and marine world, and a lot of antibiotics and anti-cancer drugs which are in the present market are derived from marine world. Similarly, some of the drugs also discovered from venomous and toxins. So, if you see applications of natural products are applications are everywhere, starting from food to pharmaceutical application, pesticides, cosmetics, and herbal medicines like so and so. Here I gave some of the best examples. Here you can see the artemisinin. We are very much aware that the discovery of artemisinin was rewarded Nobel Prize to Chinese scientist in 2018 or 17 so. So one of the best medicine even till date, there is no replacement by synthetic drugs, artemisinin, and ivermectin, which is a very well known as a craprodactin, and rosmarinic acid, and tropoxin B, and applicant epigenin, which is a very widely used as a colorant in the beverage industry, and the linalool is very well known in perfume industry, and stereoside is very much used by diabetic people, non-sweetener, and then linoleic acid is a lotion, widely used in the lotion industry. So these are few examples. There are a lot of examples in the market where there are only the blockbuster drugs which are derived from the natural products and applicable to the many sectors. So if you see the beauty of the nature here, see the plus limonene, the plus isomer and minus isomer. Plus isomer gives orange smell and the minus isomer gives lemon smell. Similarly, if you see the peppermint and minus is a carbon is a major compound, minus isomer gives spearmint smell and plus carbon other isomer gives carbon smell. This is one of the best example on beauty of the nature in the section of the chemistry and isomer. One of you, I think you must have known about this QS21, which is a natural products based saponin and widely used as a adjuvant in different vaccines, including our Covishield vaccine. And these are only the adjuvant natural products based adjuvant, widely there is no synthetic. The only source is natural products. And if you see, as just now that our scientist mentioned about the spices. So even spices also contributed a lot of health benefits in the particular Indian tradition. And recently, with the COVID, in the COVID-1 or COVID-2 period, one university, Savitribai Phule University and D.Y. Patil University, they have conducted a survey regarding the consumption of consumption of spices versus viral load. So they have conducted a survey starting from Kashmir to Kanyakumari and the Lakshadweep Island and Andaman Nicobar Islands and other islands also. So what they found, the people who consume a lot of spices, they are much immune when compared to the other people who do not consume the spices. So that's what in the Andaman and the Lakshadweep people have, there is a minimal mortality rate during COVID when compared to the mainland people, because they have the specialized, like they cultivate specialized spices in the island. That's one. So this result shows really eye-opening results for particularly chemician pharmacologists. Our spices contains the polyphenolic compounds which are having the properties of adjuvants. As you know, adjuvant is nothing but the compound which increases the immunity response. So that is so based on this observation, Indian Institute of Technology, Madras, Chennai, IIT Chennai, biologists and chemists together they did some work and then find some of the molecules which are potent or against the cancer as well as the adjuvant therapy. So they have recently they filed a patent also, and this is also one of the good areas to explore the polyphenolic compounds or anthocyanins or proanthocyanins which are having a very good potential to develop as a adjuvant therapy as well as anti-cancer therapy. So why happens? Because natural products has a history of the utilization since way back in the Mesopotamian civilization, way back 2600 BC, which has been, there was evidence utilized by the Mesopotamian civilization. Similarly, in the Egypt civilization, 1500 BC, in that time, at that time, 700 drugs were documented. You can see how they are much forward when compared to our civilization. And coming to the Chinese medieval medical, which is a very famous system of medicine, which is traced back to 100 BC, that is also one of the biggest system, like our Indian system of medicine. System of medicine of each country, particularly our Ayurveda. Ayurveda, as we know, it is a long established system of medicine. Medicine. Medicine like extracts and congress and decoctions. A lot of base for the market. Here I several modern molecules or several promising molecules have developed protein and performed the Ayurvedic art. Here all the Ayurvedas which are derived from Maurya, Sapindra, Surya is derived from Swaraya and Rola some examples, Pico's philosophy, curcumin, and steroid glycosates, which are very common immunomodulators derived from Ithania somnifera. These are all economically affordable, that means the plants are easily available, as well as some of the plants are cultivated by the farmers, and procurement of raw materials also very easy, and they are compared to low toxic and very effective in chronic diseases. So toxicity, coming to toxicity, these plant-based drugs and Ayurvedic drugs should take the optimum level. Dosage should be optimum, and it should be taken by the advice of the Ayurvedic prescriptions. Otherwise, there is also very adverse effects also there. So I will tell you an example. One of my friend suffering from diabetes, and he's working as a lecturer in government college, and his colleague told how he has shared some suffering from diabetes. Like his friend told, why don't you take Strychnos naxomica bark? Because Strychnos naxomica is a very vital plant. Its bark is known for its diabetic principles, and its fruits are toxic, and which will be mixed in the monocrotophos and applied to the paddy fields and as a pesticide. So naxomica capsules are also very famous in homeopathy for diabetes. So my friend, what else, you take some of the bark and put it in a glass of water. Next day morning you take that water. That's nothing but a water extract. So my friend was so excited, and he has taken more bark. So the dosage is very high. So he was hospitalized because of if you take more dosage, our body cannot accept the motions or vomitings. So if you take optimum dosage, definitely there is no side effects, and it works very well, particularly chronic diseases. That is the main the advantage and disadvantage is only the organ kidney will be damaged, no other organ will be affected. That is one of the biggest disadvantage of consuming herbal drugs. If you take more dosage, the organ kidney will be damaged. So if you see recent Nobel Prize winners in 2018 and 2016, in 2016 the Nobel Prize winner related to the discovery of mechanism of autophagy, it is almost indirectly related to our Ayurveda fasting. Fasting. Similarly, in 2018 for the cancer therapy, this is also related to indirectly our Vata Pitta Kapha Ayurvedic concept. So that means our travel system is in place, but we are not much using, and we are because of there are several reasons. I can share some of the reasons that if you see the biodiversity, and there are 34 megadiversity hotspots in the globally, and our country hosts 3 regions. One is Indo-Tibetan region, Himalayan region, and Western Ghats. In these 3 regions, there are a lot of medicinal plants which are used by the tribal people, particularly in the northeast region. Almost 80% of the people, they use the folklore medicine from the surrounding medicinal plants, and some of the medicinal plants they are not even characterized taxonomically, but they are using and they are benefiting a lot. and some of the medicine they are exporting to other countries. So, and if you see the herbal technology, India still almost around 8,000 to 10,000 medicine species are they are categorized like pesticides, gums and resins, fiber plants, and other importance, and then edible plants. So, if you see the contribution of the herbal technology in our system of medicine like Ayurveda, Unani, Siddha, Amji and modern medicine, and almost close to 1,000 species is very interesting in the system of medicine, and in modern medicine also nowadays, after the cancer patients, particularly after having chemotherapy, now doctors are advising plant-based nutraceuticals for taking the cancer patients instead of giving the tablets. So if you see the contribution from the last 4 decades, almost more than 2,000 drugs were marketed, either pure natural products or its derivative. So only 80% of only 20% of the Indian flora has been explored so far. Remaining 80% is as it is. And when coming to fungi, more than 3 lakh fungi is available, but only less than 1% is studied. So this is also one of the good opportunity to work on in this area. And if you see the contribution, more contribution is in the area of anti-cancer drugs and anti-hypertensive drugs, antibacterial drugs. So even till today, amphotericin B, which is called as a gold standard, is sourced from the natural products, and the lipopeptide, caspofungin, which is one of the famous drug for antifungal therapy, is also derived from natural products. Even for dengue therapy, doctors who are prescribed the papaya leaf extract, that is nothing but Siddha medicine. So even till today there is no medicine for dengue virus. Only thing is the the indirect therapy, increasing the platelets so that it will automatically fight against the virus. That is one of the best advice. Even till today there is even majority of the companies are preparing the papaya leaf extract and selling in the medical shops. So that is a good thing. Likewise, several other drugs also, only there is a source natural products. Here, if you can see the categorization, it is like a simple, when you buy some of the things in the roadside kirana shops, price will be less. If you buy in the supermarket, price will be high. That means there is a difference in the quality. Likewise here, we can categorize herbal medicines in the 4 categories. Category 1, herbal community, regional folklore medicines where there is no scientific sanctity, there is no like a treatise or a pharmaceutical for the preparation of this medicine. Category 2 comes under Ayurveda and other medicines where those are prepared by following some of the documents like Ayurvedic pharmacopoeia and other pharmacopoeia. Category 3 comes under modified herbal medicines and formulation. So some of the quality and efficacy data required. And category 4 comes under completely imported medicines where there is a full efficacy, safety data, and all data should be there. Then only that will fall under the category 4. So if you see a global scenario, more than even the underdeveloped countries also using the plant-based medicine, herbal medicines, more than 50% of the people. If you see Australia, USA, France, Canada and Germany, but name is different but raw material is same. Why the plant-based medicines are working, what is there? Plus plants contains the primary metabolites and secondary metabolites. So primary metabolites are the chemical substance which is required for plant itself to further grow, and almost very less than 1% primary metabolites also used in the formulations. And the examples are proteins, carbohydrates, lipids and nucleic acids. When you see the secondary metabolites, these are the real compounds where these compounds are evolved to the defense mechanism pathway, and they are the real heroes to fight against the different diseases. And we can differentiate these secondary metabolites to nitrogen containing compounds, polyphenolic compounds, terpenes and all other different categories based on the structures of these compounds. Alkalize, nitrogen containing compounds means alkalize, nicotine, morphine, cocaine, caffeine, glucose, phenolic compounds, where hydroxylated compounds, anthocyanins, salicylic acid, lignans, tannins, salicylic acid, and terpenes, monoterpene, diterpenes. So there are different categories where these are formed through the circulation of the isoprene units. And if you see the evolution techniques in the natural products in 1800, there is no much scientific advances. So one isolation, characterization, pharmacology, even techniques are also very immediately available. So keep on coming to 1900th century, 1900. So some techniques are being evolved in the characterization of the compounds and even PLC chromatography, UV visible spectroscopy, and then now in the era of 2025 we have a lot of even spectroscopic techniques and biological high-throughput screening techniques, X-ray, whatnot, even NMR 700, 800 is also available. So this is the evaluation techniques. And if you see the success history in 1800s, at that time people used to take whole extract or fraction instead of taking the single compound medicine as a drug. So at that time morphine was the first compound where it was isolated, and it is also known, proportionally is also known as a traditional medicine. From there they have used a biocellular approach to isolate the morphine, which has later become a drug. That is the first compound which has become a drug from traditional medicine, from discovery to delivery. So based on that, several compounds were discovered, and then later that also became a drug. And first semicynthetic drug was discovered in 1899 by Bayer. When coming to the milestones in the discovery of penicillin, it's very well known, 1920 from penicillin notatum, and after that several drugs, antibiotics also discovered in 1940 to 1970. Best examples are tetracycline, streptomycin, doxycycline and lipopeptides. This 30 years period is called golden era of antibiotics, and intensive investigation of the marine environment began in 1970s onwards. After that, several marine anti-cancer drugs, antibiotics were discovered, which are in the present market. And this is the scheme of the discovery of natural products, starting from bio-raw material to the drug candidate. So here there are different steps involved, starting from bio-raw material to drug candidate. First step is bio-raw material, which may be plant, fungi, insect, microbes and marine, and that will be processed for getting the bioactive extract, which will be there are different extraction techniques, oxalate, counter-current chromatography, solid phase, supercritical carbon dioxide chromatography are for extraction. So based on the compound nature, the extraction techniques will be adapted. Once the extract is prepared, then we need to identify pure compound. I said the pure compound by different chromatographic approaches. Here also there are different chromatographic approaches available. So based on the compound polarity and compound nature, these chromatographic techniques will be adapted, like HPLC, counter-current chromatography. And once compound is purified, then it has to be characterized by using the applications of different spectroscopy techniques, starting from UV visible, FTIR, NMR, MS and X-ray. And once the compound is characterized active, then that has to be prepared in larger quantities, which comes on medicinal chemistry will come in this picture, and then once it is prepared, it has to go through the different phase studies to become a drug candidate. Almost this will take 10 to 15 years from start to step 1 to step 3 years. That is what nowadays even many pharmaceutical companies are not coming forward to take the drug discovery from the natural sources, but they are taking directly one lead compound and they are approaching the medicinal chemistry approach for developing a drugs. So there are the main key steps in the natural drug discovery: identifying the hit to lead and lead optimization. And there are some issues like biodiversity is one of the issue, and patentability and patent protection also one of the issue. I think we might have much aware regarding the fighting between USA and our country for getting the patent back of our Ayurveda indica, neem. So actually America wants a patent rights, but that is a completely Indian origin, particularly Asian origin plan. Particularly CSI fought like anything, and the patent got back by our Indian community. Similarly, there are other communities, Tulsi and there Curcuma is also one of the best example. So Curcuma langa and Ayurveda indica, these two are Indian originated plants, particularly those are part of our tradition. So they cannot steal. So likewise, there are a lot of issues in the natural products, and it is and there are some of the research areas also in the natural products today, particularly metabolomics and synergy and biotransformation and biological activity, molecular biology and biotechnology, quantitative natural chemistry based on the analysis. Particularly, the natural product activity is a multidisciplinary activity based on the folklore knowledge is one of the base. It is a kind of getting the validation of the folklore medicine or traditional medicine. What is the candidate, what is the compound which is in the present in the crude extract or folklore medicine. It is like identification and validation by the biases. That's what always folklore healers and Ayurveda, Ayush, these are the main backbone. Driving force, and based on the taxonomy, cancer biology, bioinformatics, ethnobotany, these are all it's like a multidisciplinary activity for drug discovery. These are some of the thrust areas in the traditional medicine, metabolomics, phytoequivalence, phytoinformatics, and reverse pharmacology, one of the approach, and safety evolution, and globalization of the traditional medicine, herbal pharmacovigilance. These are one of the latest thrust areas in traditional medicine. And just I will give a small glimpse about the extraction and analytical techniques in the natural products. So as I mentioned in the drug discovery process, there are different extraction techniques starting from succinct extraction, maceration, supercritical extraction, refluxing, hydrodistillation, and spectroscopic technique, NMR, IR and UV, and 2D NMR is also one of the crucial technique for the identification of the structure. NMR gives a number of protons, proton NMR particularly gives impression about the number of protons present in our sample and the number of carbons which will give proton carbon 30 NMR. 2D dimensional NMR gives information about the carbon-carbon linkage, carbon-proton linkage. And coming to the mass, there are different mass spectrometries like based on the compound nature, the analysis techniques will be identified like electron impact mass spectrometry and a far mass and molecular ESI technique based on molecular weight based on ionization techniques. So these are the main approaches in the natural drug discovery. Biocide approach and reverse biocide approach or random approach, and NMR based or mass based approach is also called metabolomics based approach or delignification techniques, and metabolomic based approach and molecular networking based approach. The last approach is almost related to AML based related kind of approach. So in the biocide approach, the active extract will be taken and further it will be fractionated by eluting some of the solvents, and then the Indian fractions will be screened for different targets. The active fraction will be taken up for further the isolation of the compound. Once compound is identified, again its target validation can be done. And when coming to second approach, reverse biocide approach or random approach, where there is no need of following by every step in the bioassay, just you isolate as many compounds you can, and later just screen all the compounds against the target, then you can find some compound. Because sometimes what happens is your active compound is minor in nature and inactive compound is major in quantity. Sometimes the active compound will be masked by the major compound which is inactive. So in that case you can miss the reactive compound. That's what the secondary approach is also equal approach. And NMR and mass based approaches are evolved for the delignification, which really saves the time and accuracy. And molecular networking approach is a approach where it's an artificial intelligence based approach. So there are different thrust areas for startup opportunities, process development of active phytochemical ingredients. As we know, the drugs vinblastine, vincristine. So far, Cipla is a monopoly. There is a lot of requirement in the market. So that is one of the biggest opportunities now. And similarly, herbal nutraceuticals, functional foods and dietary supplements is also big market in the present scenario. And really, the herbal supplement is no side effects and exhibits robust growth, major factors and rising consumer prices. And when you see the market analysis, there is a big rise in the plant-based market. Traditional Medicine Act 2014 to 23. And here I give some of the examples derived from terrestrial plant. The first one is artemisinin, which is a very famous drug against anti-malaria. And as you know, this is also one of the traditional medicine. And based on the traditional medicine plant, artemisinin was isolated and discovered by the Professor Ever, is lady scientist from China. And this was actually there was a war between Vietnam and China. The Vietnam scientists were majority of the soldiers from Vietnam was passed away during the mosquito bite not by the attack of the opponent. So in that case, the Vietnam approached the China because Chinese system of medicine is a very longest and oldest system and popular system. So at that time, China government has constituted a committee under the chairmanship of this lady, and they have screened almost 400 to 500 medicinal plants which are very much used as an anti-malarial treatment in their system of medicine. And then finally they end up with the one plant Artemisia innova, and then from there they have isolated Artemisia under the cold temperature conditions. So this is a background story of discovery of Artemisinin. Based on the discovery, several other drugs also been added. So by doing the simplest modification like ethylation and methylation, and by adding the fluorine group, so these drugs also enhance the activity and also selective malarial treatment, and these drugs also approved by the USDA and USFDA for the treatment of the anti-malaria, and they are in the present market for the treatment of anti-malaria. Similarly, there is another drug called Taxol, which brand name is factory Taxol. So this was Taxol was isolated from the Taxus brevifolia plant in 1967, and its mitotic activity was discovered in 1967 only, and when it is commercialized to Bristol Myers Squibb, it was named as a factory Taxol, and then later it was got a license for different cancers starting from lung cancer, breast cancer, neck cancer, all other cancers. And also this is also approved in different cancers as a combination drug, as a single drug in different years. So this is a semi-synthesis of Taxol, because Taxol is a low yielding compound in the plant, and however this fragment 10-diacetyl bicarbonate is a major compound. So this synthetic tail used to enclose this major compound 10-diacetyl bicarbonate. So far this is the only synthetic procedure for the synthesis of Taxol in the ton level globally. And here also there is a lot of opportunity for startup. And Taxol, even till today, there are many cancers are diagnosed. Even Taxol, Vinblastine, Vincristin, they are working very well, and there is no sufficient compound available for the patients. So this is also one of the startup thrust area. And Vincristin and Vinblastine, as I mentioned you, these are isolated from Catharanthus roseus, and this plant is also a traditionally identified plant for different treatments including cancer, and these two alkaloids are isolated from this plant and widely known for chemotherapy, and they are approved by the FDA for the treatment of cancer 1963 and 1965. Later they came to know these two compounds are working as the inhibition of the tubulin polymerization and beta-tubulin heterodimers, and preventing the microtubulin polymers. And this is also later several synthetic drugs also being developed from the Vincristin and Vinblastine by doing the simplest modification. First synthetic is a Vindocin, and they are just further it is also approved for treatment of lung carcinoma and breast cancer. And Vinblastine, it is also another synthetic drug approved in 1989 and approved for breast cancer. And by adding some of the fluorine group CF3, they have also developed one drug, Vinfluorine. It is also approved in the market 2009 for different cancers. And this is a global market for Vinca alkaloids. You can see 2022-23, there is a lot of escalation. As I mentioned you, only supply is a monopoly. There is a lot of opportunity. Similarly, there is a lot of complexity also in isolation and process development of these two alkaloids. So there are other drugs, camptothecin and fluorophyllotoxin, where it is also isolated from the different plants. Camptothecin acuminata, which is a Chinese medicinal plant, and it is a quinoline alkaloid, and fluorophyllotoxin isolated from Fluorophyllum peltatum. And based on this, several two drugs, etoposide and tenofoside drugs also developed by adding some of the glycosides and thiophil moiety. And these two important drugs also approved by the 1996 by FDA for different cancers. So if you see some of the quality control aspects, here this is a paper clipping from August 1st, 2023, saying that herbal supplement toxicity lands patient in the hospital. what happened is because of this adulteration in the herbal drugs, because of the curing, she affected by the toxicity and adulteration and she was admitted in the hospital. So that's what there is a lot of quality control issues in the market and there is a lot of adulteration. Otherwise, we are really blessed with nature flora for different diseases. And why? What are the major problems in the Indian herbal? Because of there are different regulatory affairs. See, I share some of the guidelines for getting the drug into the market, and another agency says different drug. So finally, herbal industries are following the shortest route to overpass some of the guidelines, and they are getting the market. Finally, there is no efficacy and toxic studies also not being done while getting into the market. That is a major problem. And in some cases, if the major plant is not available, they are also using the plant which is synonymous to that plant, where there is no studies. Studies were done that synonymous plant. That is also one of the major problem in the present scenario. The therapeutic effect of the medicinal plants are due to the presence of the biologically active compound, which is called therapeutic marker. So that may be all secondary metabolites like alkaloids, glycosides, and almost 70 to 80% of the medicinal plants are sourced from the wild source, because there is a different fingerprinting profile or different parameters of different proportions of compounds from wild source and cultivation source. It is like a pampered chain and self-grown chain. You know the difference between these two. Like this, so but while doing the formulation, you need to optimize all the compound, presence of the compounds and fingerprinting profile and profile of the plant of the season also you need to identify, because the plant profiles are different when you compare which is not and temperature is very low, chemistry is the plant becomes prepare the type of compound, different type of compounds. That's what some of the companies, like for example, Dabur Meswak plant was in Nepal, where my ingredients those are very good enough with market compounds in the plants available in the Himalayan region, Nepal region. That was the manufacturing plant was done in the Nepal. So likewise, we need to follow some of the SOPs and we need to optimize each and every stage and quantify the active markers in the formulation in the formulation for the presence of the active markers. So these are some of the standardization parameters and quality, quantity, observation and efficacy. Here comes to this, has to be checked with the parameters while doing the standardization of the herbal medicines, like physical parameter, chemical, biological and botanical. In the physical parameters, we should check the moisture value, extractive value and chemical. The plant raw material should not be contamination of pesticide and heavy metal, and biological should not be a microbial contamination, and toxicology, pharmacology evolution should be done. In the botanical, it should be thoroughly checked macroscopic and microscopic examination whether it is a genuine plant or the adulteration of synonymous of the plant. So these are the different techniques in the standardization. These are the different hyphenated techniques approved by different agencies like HPLC, HPDLC and UPLC, LCMS and GCMS. So every technique has its own advantage in the characterization and standardization of the herbal medicines. So this is one of the examples, as we are also working in this area for quality control aspects of different herbal drugs. So this plant is Sassurya Lappa, widely used plant for different herbal formulations. So these two are the major compounds like castor oil and epidihydrocastor oil. So when you use this plant, these two compounds must be there. Then only those formulation is genuine, otherwise it is adulterated. So based on this, we have procured the raw material and then we have quantified these two markers in this raw material by developing the HPLC method. And further, we also purchased some of the marketed herbal formulations where this plant is one of the ingredient and then GSC, the flaxseed and CC surrounded by the mountain and then sea between the mountains. That also matters for the biodiversity of the marine environment. So generally, the majority of the marine products have been isolated from sponges, coloncates and tunicates, echinoderms, bryozoans, a wide variety of microorganisms in their tissues. And these are some of the pictorial representation of the sponges, molluscus and tunicate and bryozoa. As I mentioned you, the marine is the biggest source and there is a lot of animals, very small fish to very big animals, and always there is a competition for the survival of each and every animal. So in that process, every animal will protect themselves while if it is attacked by the other animals. In that process, the animal or organism will release some type of chemicals to protect itself. That is called the defense mechanism pathway. That is what majority of the anti-cancer or antibiotics are derived from marine source. There is one example. There is a called sea cucumber. It travels only 100 meters in a day. When it is attacked by the big animals, so it will release instantly some kind of spray to protect itself. That is nothing but the defensive mechanism pathway. In that process, immediately, spontaneously, it will release some kind of weapons. So the marine sources, traditional sources, plants, and high level of biodiversity in the marine world. High level of biodiversity means high level of chemical diversity, and the complexity and the variety of molecules and toxicity molecules are very high, and which are nothing but potent new bioactive molecules. As I mentioned, there is a competition for survival and environment pressure in the sea, which leads to the biodiversity and biodiversity. As I mentioned, defense attack signaling. They develop some of the chemical compounds, there is a chemical diversity, which finally leads to a potential new drugs. So, this is one of the drug, giconotide, which is isolated from the snail. This is the first drug, marine drug, originated in December, approved by the FDA in 2004. So, this is later came to know this blocks the entire calcium channels and the primary nociceptive nerves in the spinal cord, and it is also one of the most powerful painkiller to date. And this is a holychondrin B, which is isolated from the sponge. Holychondrin occurred in 1985. From this drug, since there is synthetic modification by extrapolation of drug, the ergotamine drug was developed by chemical modification. Finally, this ergotamine was the most potent drug till date, and this is also the present market for the trade name of Hyaluron injection for the breast cancer treatment. So this is a only drug which works very well to treat for the breast cancer. And another drug is Bryostatin 1, which is a microlactin isolated from Rubella erythema, and this is also approved by different agencies for the cytotoxicity for melanoma and kidney cancer and lymphoma cancer. These are all examples of several other drugs, anti-cancer, antifungal, and the antiviral drugs derived from marine sources. And as I mentioned, animal derived secondary metabolites, the epigadrins and the capropil is two of the drugs isolated from snake venom as well as the frog. These are in the present market. Another source is lichens, a small source. Lichens are nothing but symbiotic organism of fungi and cyanobacteria. That is a friendship, growing friendly between cyanobacteria and fungus. And they are very nutritious lichens because majority of the lichens are edible in nature. They are having polyphenolic compounds which are very good for our health, like antioxidant and other health benefits. And if you buy biryani material in Sunday, the biryani pack contains lichens. So that is one of the best example. And even if you visit some of the tribal people, they every Sunday they mix this lichen in the vegetable they eat. So so far there is no drug derived from the lichens, but lichens are added to the different other drugs as an additives for particularly cosmetic drugs. So lichens are grown particularly in the moist regions and non-polluted regions, and they are also known as the pollution indicators where they grow in the non-polluted areas. And this grow in the eastern region and western region, particularly. If you see the lichen diversity, Tamil Nadu is the richest state, followed by Uttarakhand, West Bengal, Sikkim, and other states. So another natural place is seaweeds. You must have heard these are also called macroalgae. Macroalgae. This green algae is edible, which is rich of protein, and this is like a served in the Japan and other countries like a salad. So very rich protein, and red algae they use isolated from the seed. This is only of also isolated from the image. Food, animal, biofertilizers, biopolymers, nutraceutical, cosmetic, industrial chemicals, and CO2 and this is a method of cultivation which in government of India also gave support for the seaweed cultivation. And these are the different states cultivating, particularly coastal states, Maharashtra, Gujarat, Tamil Nadu, Karnataka, all states are cultivating. And still these are the different uses of seaweeds and nutraceuticals, cosmetics and food supplements, biofuels, biofertilizers, growth stimulators. And even ice creams also use seaweeds. So when coming to our own research, we have been working in this area from the last 20 years and we isolated several compounds, active compounds, and some majority of them are patented, and some of our drugs also in the present market as a nutraceutical, as a drugs. And we work, generally we visit the interior areas and interact with the tribal people to understand their knowledge, and coming to the lab we will by different databases, whether if those plants are not validated by science, then we will take up the challenge and isolate the fraction and isolate the compound and characterize, we screen for different targets. On the industrial side, we are working on the standardization of the multi-herbal formulation by with the help of different sponsorship of industries and pilot scale of compound process development. And this is a new discovery process at ISCT. And this is our approach where we do natural products isolation as well as medicinal chemistry of different marine sources, plants and system of medicine plants and biological activity, and if they find very active compounds, we will go for a patenting and later it will be developed as a drug candidates. So yeah, I conclude here. I would like to thank once again for the organizing committee and management for giving the opportunity. And I thank my director and my supervisor Dr. Jain Rao and for other funding agencies, and not least my students for their hard work. And here I would like to, I'm happy to share two of my drugs are in the present market where we have developed some of the compounds as a alpha glucose inhibitor as well as some other activities. And we filed a patent, US patent and European patent. Later these two patents were commercialized to Sami Labs Bangalore. Now they worked for some time and then released as a nootropic agents, diabetic related neuropathy in the USA market in the name of Saproxy tablets and Saproxy syrup for cognitive compounds. And this is a trade name. And these are the Saproxy tablets 100 mg, Saproxy powder for nootropics. And we also work on the nutraceuticals or nutrafood. We developed two formulations for particularly anemia and then diabetes. And these two technologies were taken by Amawala Sciences Hyderabad, where they made a product in the name of superfoods for you, different snack item, and also mixed with our wheat powder, diabetic atta and atta. And this product was also launched in 2023 in ISCT by our Honorable Science Minister Jitendra Singh Ji. And this is a demonstration of the process and of the like a kurkure, like a edible item process. And this is an exchange of technology. And this is a release of the product in our auditorium during the visit of our Science Minister. So just I would like to take few minutes for the description of our organization. So CSR IOCD comes under the Council of Scientific and Industrial Network.
All students, please note that sir is giving the introduction of CSIR, so please listen carefully.
Okay, CSIR comes under Ministry of Science and Technology. CSIR has almost 37 laboratories in the entire country in the disciplines of biological sciences, chemical sciences, engineering sciences, information sciences and physical sciences. Hyderabad, the color shows the discipline. Hyderabad has 3 laboratories, CCMB, NGRI and ISCT. ISCT comes under chemical sciences, CCMB biological sciences, NGRI physical sciences. Here NCL, National Chemical Laboratory Pune comes under biological sciences. So when coming to ISCT was established before India's independence, 1944. For the last 80 years, ISCT is serving in the for the humankind and supporting the herbal industries as well as pharma industries. And we have public goods, private goods and societal goods, different sectors we are working. Our strength is major. We have 150 plus scientific staff and 300 plus supporting staff, 500 to 600 research scholars are working. And we have around 1700 patents registered and every in India and abroad. And so far more than 500 PhDs were awarded. And research publications we get on an average 450 or 400 research papers every year. So this is the overall glorious year of CSIR, and we have different research areas starting from applied biology and chemical engineering, process chemistry, fluoroorganics, polymers and organic synthesis and process chemistry, natural process and medicinal chemistry, where I work in this division, catalysis and fine chemicals, energy and environmental engineering, oils and lipids, and analytical chemistry division. So we work in the healthcare sector, chemicals, environmental industry and energy. And these are the services. We also offer different services like R&D projects, analytical service also we do, and process engineering and applied biology. We have state-of-the-art analytical facilities. We have 8 NMR machines and almost 10 to 15 mass LCMS machines and everything. And these are the recent technologies we have now, success stories are from CSIR, ISCT, AGR technology, anaerobic gas reaction technology, from waste to gas. And this is also quoted by Prime Minister Narendra Modi ji in his Man Ki Baat. This gas will be generated from the food waste and vegetable waste from our canteen, as well as we are establishing this same plant in the vegetable market, Boyampalli. And this is we are also working on the membrane filtration technology. We have established the different plants in Mumbai railway station and Secunderabad railway station, where water will be produced from the atmospheric moisture. And we also installed several plants in the fluoride affecting areas of Nalgonda, Telangana and Andhra Pradesh, and of course in the government schools. And we also contributed for COVID-19 solutions like Covaxin to Covaxin, where one of the compound adjuvant, the processor developed by the ISCT and given to the Bharat Biotech Industries. That means every Covaxin well contains ISCT technology, and developed a process for favipiravir and other colchicine-based drugs also developed a process and licensed to Laxai Industries, Hyderabad. So I'll continue here, and thank you once again for your patience, and I will be happy to answer any questions.
Thank you so much, sir. Now the session is open for question and answer.
Sir, good afternoon, sir.
Thank you.
You have dealt into natural products from in-depth, like an ocean of knowledge in the source of natural products. And you have went back to the traditional medicines, that is from the plants to the marine natural sources and various compounds that have been isolated from these natural products. Sir, you have told one of your friends, you advised him to take bark of Naxomica. Yes, sir. So, seeds are poisonous, but you have advised him to take bark of Naxomica. And he has to overdosage. One question that is there. And other is for dengue fevers, dengue viruses. We are taking the leaf extract of papaya. So what chemical constituents might be responsible for curing or treating the dengue virus? Thank you.
Yeah, this Strychnos naxomica, as we know, it's a very good plant. And the strychnine alkali is one of the major compound in this. And the fruits are very toxic enough. And the bark is having medicinal property. Naxomica capsules are known in homeopathy, made from the bark. So they are using the fraction or extract, not the single compound, but the plant majority contains alkaloids. Strychnine is a very well-known alkaloid. When coming to papaya leaf extract is known for its platelet aggregation. which is very well known as the Siddha medicine. So majority of the times plants are plant-based therapies work in a synergistic pathway. Like all crude extracts are the fractions. So maybe that is also containing alkyls, and majority of the work has also been done to isolate the active compound, but that is not working on par with the extract which we active. That's what the majority companies are using the extract only, not the simplified compounds or fractions. That is one of the reason. Thank you.
Sir, just I have one question. So nowadays most of the people they are taking the allopathic medicine and they are not using the herbal based the roots or leaves. So can you give any suggestion regarding this? They are completely avoiding the herbal medicine and leaves extracts like that. Completely they are using allopathic.
It is not like that. See, it's completely depend upon the individual interest and then other things. And see, after COVID, now people are more interested to use the more spices particularly. You can see a lot of ginger we are consuming daily in our, starting from tea to in curries and all those things. So that is one of the change. And even roadside also you can see number of teas like ginger tea, ginger jaggery tea, and even cinnamon tea, lot of spices. And that shows the trend has been slowly moved to change. And another thing is, as it is my personal opinion, see, when COVID-19 attacks, our country like a big country almost populated with 140 crores population. So do you think we will really survive? That only our gift is our food or our tradition, like consuming spices that inherently increase our immunity that came from our ancestors. So that is the only medicine, that is the only mantra which really saved us. That is my personal opinion. So we have the inherent immunity based upon our traditional food habits and like this. And really the people now more conscious and more interested even using plant-based medicine and more interested even on the organic based foods. People are like, if you say organic tamarind, people are made a big queue for purchase. See, tamarind is a wild, there's no pesticides, nothing. Even then, if you say organic, people give 100 rupees more and they will buy. So that shows the transformation. And Ayurvedic is also, slowly people are changing, and there is a lot of potential in our traditional medicine like Ayurveda and other things. And I have also seen personally, and because of the adulteration and not making properly by proper guidelines and by proving the efficacies and toxicity, those were not becoming famous. Otherwise, see, if you see, there is no base. Even lot of whatever the medicine which are in the present market, the base is nature. Nature is like our mother. If you really use properly, definitely it will protect us. If you really spoil nature, definitely will face. That is what the natural calamities are happening. Because of pollution, utilization of the plastic, really we are facing. See, a lot of problems, incidence of cancer, see, temperature rise and all those things. See, unexpected floods. And then we have you heard of any, like a cloud burst, anything like before 10 years? No. Like sun burst, cloud burst, these are all, because these are all man-made things. And best example is Shalaki, which is a very well known of the arthritis pain. Boswellia serrata is a major plant. So there are 6 compounds very well known in that market, and we have recently developed a process of isolation and quantification of that one, and we have purchased almost 40 to 50 formulations from the market of different multi-herbal formulations, cream, oil and everything. Only 2 to 3 companies are following the same guidelines. Other more than 35 companies there is no guidelines, there is no toxicity, and also in some companies there is no even one compound, only jelly. So this is a major fate. So otherwise there is a lot of potential. Thank you. Now people are shifting. Any questions from students?
Please raise your hands if you have any questions. So, No one is having the question, so I conclude that question and answer session is over. Thank you so much, sir, for this wonderful and informative session. So next session will be of Dr. M. Shankaraya, sir. So I request Alpha ma'am to introduce Shankaraya, sir. प्लीज ऑल पार्टिसिपेंट स्टे हिअर. सगळ्यांनी इथंच थांबायचं आहे. नेक्स्ट सेशन लगेच चालू होणार आहे. कोणीही ऑडिटोरियमच्या बाहेर जायचं नाही. सेशन लवकर संपणार आहे. इथेच बसायचं आहे सर्वांनी.
Good afternoon. Dr. N. Shankaraya, sir. Welcome, sir. Sir is currently working as a professor in Medicinal Chemistry Department and the NIPR Hyderabad, NIPR Hyderabad. Sir has completed graduation and post-graduation from Kakatiya University, Warangal. PhD in 2007 at the CSIR, Indian Institute of Medical Technology. Sir has completed post-doc from the University of Talca, Brazil. Sir is having more than 25 years of teaching and research experience. Sir is listed as among the topmost 2 scientists worldwide for the last year according to the Elsewhere. Sir research focuses on the designing and synthesizing new chemical entities for the cancer, drug discovery and developing synthetic methodologies in pharmaceutical science. Sir has published more than 195 research articles in reputed international journal with high impact factor. Sir has contributed in various books. Sir is having more than 5 patent articles. Sir research articles having more than 6,400 citation with H index 64. Sir has supervised and guided 19 PhD students and 142 M.Pharm students. Along with that, many students are doing postdoc in reputed universities like Harvard, Washington University under Sir's guidance. Sir has delivered more than 60 invited talks at various national and international scientific platforms. Sir has elected as a fellow of Telangana Academy of Science for the year 2021. Sir has received OPTI Young Scientist Award in 2010 and Best Research Scientific Award in 2016 from NIPR Hyderabad. Sir is member of many academic bodies and governing bodies at various academic university. I think it is very short to explain, sir. But we are eagerly waiting on the topic which is sir is delivering today on the role of molecular hybridization, which is approach in cancer drug discovery. So I hand over the session to you, sir. Thank you.
So can you hear me? Right. So thank you for your kind and very short introduction. And at the outset, I must thank the organizing committee for inviting me, especially management, Dr. Vishal, and then Srikanth Srinivas, who has been conducting from more than 1 month to materialize this conference. It's a great, I came to know that more than 1,500 registrations happened for this conference. So it's not easy to organize a conference. So I think it's going from last 2 days successfully, and many students are very enthusiastic to run the these all whatever the talks are going on is like out of your textbooks, right? So now it's 12:20, so everybody is like eagerly waiting for lunch. Okay, I will try to finish the earliest, so please concentrate. the topic. So I'm going to talk about medicinal chemistry and basics of drug-like properties and how new chemical entities they are talking on small molecules, how we can design small molecules. And when you are designing small molecules, what are the parameters you have to keep in mind as a medicinal chemist or synthetic chemist? Okay, so please concentrate on this topic. So I am going to take today the role of molecular hybridization in cancer drug discovery. Molecular hybridization, you know all of you hybridization, we studied, right? Hybridization. Here is a molecular hybridization, means you are combining two pharmacophores or three pharmacophores and making a single entity. Okay, so the first talk has given by Dr. Heather about multi-drug resistance, you also know. So why that multi-drug resistance has came, and in that how this molecular hybridization is helping in the design of new chemical entities. So this is about my outline of today talk, the discovery and development, drug-like properties, targets, and what are the privileged structural elements while designing the molecules. And again, in molecular hybridization, there are three categories. So there is like fused approach, merged approach, and linker-based approach. As we know, the drug discovery is a long way to find a new medicine. It takes years together and effective, right? So where the drug discovery and development starts? So it starts from disease, right? So we all faced during the COVID time, how we faced the problem, right? So we all should be proud. We are in the healthcare and life sciences sector. So we are the pharmacists, are the chemists, and are the doctors. They came forefront and saved many of the lives. We are not like a software employees like working from home. So we have to work. We have to come out of our homes. So that's why we should feel always privileged. We are in the life science sectors. So in the drug discovery and process, development process is a long way to find a new medicine. So where it starts from disease. What is mean by disease? What is mean by disease? Definition of disease. The disease is a abnormal condition caused by a pathogen or bacteria or viruses or genetic disorders, not as like external effects. Okay. So the disease, from disease like a who are the responsible for the disease, and again you have to go for target identification, right? For the target you have to extract the protein who is responsible for the disease, and for that there are many people has to involve, and then they have to what is the target and how we can design molecules based on the target. We should understand the protein structure, three-dimensional structure of the protein, and what are the receptors are there, what are the enzymes are there. So what are the proteins are there, who are the responsible for the disease. So based on that you have to design molecules, and then so based on computer design you have to design millions of compounds. From that you have to identify lead compounds. So based on the lead compounds you have to synthesize thousands of compounds, and from that then you have to submit for the biological efficacy. So then you have to submit for pharmacology department or biochemistry department. They will do on cell-based assays, in vitro conditions. So they will see the efficacy of the molecules. If that they are not perfect, or if you found some of the new molecules, so then again based on that molecule you will design a number of compounds, and again you will synthesize. So then in vitro conditions, then the same compounds have to evaluate in the in vivo conditions, right? So in the in vivo conditions, again you have to see. So some of the molecules are showing very potency in vitro conditions, they may not show in vivo conditions. So accordingly, you will see in vitro, in vivo conditions, and then you will do the preclinical studies. Then formulation chemistry, pharmaceutical discipline come into the picture, and the API, active pharmaceutical ingredient. So how to formulate the one, what is the stability of the compound? So by making tablet or capsule or injectable, syrups. So based on the stability of your compound they will design, and then preclinical and clinical, and then it will go to the new drug application. So it's like a laborious process. So to find a new molecule you have to screen millions of compounds, and at the end you will get one or two drugs, and it will take 12 to 15 years. The cost of the drug, mean cost of the drug is approximately $2.8 billion. To to get a one drug molecule. It's a very, very expensive process. That's why in India we all do like a generic basis. We are not in the front fork of like a drug discovery and development. So earlier, it's like Dr. Reddy's and Ramesh, they started a discovery program. They spent a lot of money, but they could not able to reach at least one new chemical entity, means drug molecule. And when Ramesh sold their company to Diogenes Sankyo, they started the working on antimicrobials, but they could not able to release any drug. So that's the main struggles. That's why academia and industry, they are consistently striving to understand the disease and bring forth safe medicine and effective medicine to the patients. But due to lack of productivity and accelerating R&D cost and expiring of patents and the drug withdrawal rates, so that's why it's facing a lot of hurdles for the pharmaceutical industry. That's why there is a essential collaboration between academia and the industry is very essential in the current scenario. So, chemistry, one of the Nobel laureate who received the Nobel Prize in 2002, Professor Nayori, and very shortly he quoted one of the review articles in Nature Chemistry that chemistry has a central role in science and the synthesis has a central role in chemistry. So this could be considered that where synthetic chemists or molecular chemists focus their efforts to design a new chemical entities. So now chemical synthesis has reached an extreme level of sophistication where 80% of the drugs are chiral molecules. As mentioned in natural products that Suresh Babu, even R isomer, S isomer, they are totally showing different effects and they have different odors, smell. So in the same way, even our biological system also, R and S isomer, they show different effects. You know the story of thalidomide. So one is thalidomide, another one is like a, they use for the pregnant women for sedative. But it's one isomer is sedative. So likewise, the chemistry has to play a very, very important role in the design of new chemical entities. So these are the 10 elements are very, very important, crucial. That is integrity, lipophilicity of the molecules, and solubility, permeability, and durability, druggability, metabolic stability, scalability. So these 10 elements are very, very important in the drug discovery and development. So integrity means, so when you are discovering the discovery process as a chemist, as a medicinal chemist, as a like a biochemist, or regulatory affairs, okay, and formulation, how your data you are maintaining, what is the integrity of your data, documentation, that is very, very, very important. Otherwise, US FDA never entertain substandard drugs into their market. So that's why the quality assurance, quality control, and regulatory data, and these all are very, very important about the integrity and lipophilicity. So if you have good lipophilicity of your molecules, then good solubility, then you have good permeability. So the permeability is very, very important aspect. And many of the drugs, 80% of the drugs, 90% of the drugs, they cannot cross blood-brain barrier because of poor lipophilicity and the total polar surface area of the molecule, right? So and again, druggable properties, metabolic stability, and metabolic identification. So these are like a multi-dimensional application, like potential activity and safety and chemical process, so absorption, distribution, and metabolism. So as you know, absorption, pharmacokinetic and pharmacodynamic properties. So these are very, very important in the drug discovery and development. Many of the drugs fails due to this, they are not meeting these parameters. So when you take the drug, what happens is absorption and then it will go distribution, right? So when is the distribution? So it will cross the semi-permeable membranes through the systemic circulation, right? So then it will go to the gut wall and liver, and then so systemic circulation, then it will go to the metabolic acceleration balance and reacceleration in the metabolites. So what happens when you take the drug in vitro and in vivo conditions, they are perfect, very potent, okay. But when you will take it, there is a biotransformation can take place in your gut. So then the fragmentation can take place. The drug fragmentation can take place. So that is called the metabolites, like impurities. So sometimes these metabolites is highly toxic than your drug. That's why there are a lot of side effects. So that metabolites you can identify through the mass spectrometry, LC mass spectrometry. So you can take blood sample, a tissue sample, then you can see what are the impurities, what are the toxicity is showing. So in that case also the drugs may withdrawals can take place. So when you are designing molecules, you know very well lipids rule of 5. So lipids can co-work us. They evaluated more than 2000 molecules which are entering the phase 2 clinical trials. So the drug-like molecules. So then they set up some of the rules based on the ADMET properties. So the rules are like 5 hydrogen bond donors and 10 hydrogen bond acceptors. The molecular weight should not be 500 and the last value should be less than 5. Octanol water partition coefficient. And the rotatable bonds, CC or CN, rotatable bonds should not be more than 10. And more importantly, polar surface area of the molecule. So if you have drug-like molecule, the polar surface area of the molecule should be 120 to 140 angstrom square. It's like a drug. And if your molecule should cross the blood-brain barrier, it should be like 80 to 90 angstrom square, the total polar surface area of the molecule. Then only it can cross the blood-brain barrier. So that's why many of the neurological disorders will take a lot of time to cure because many of the drugs, lipopolicity is less and they cannot cross the blood-brain barrier. And the flatness of the molecule and the lab P values, polar surface area, these all play very important role in the drug design. So this is one of the molecules, like a normal example, Aeroside. So it is used by injection into the by mouth. So it is like a very good antibiotic. So it's a small molecule. So if you see molecule weight 337. So this molecule is perfectly following Lipinski rule of 5. So as a number of hydrogen bond donors, you can see acceptors and number of rotatable bonds also you can see here. Rotatable bonds, here rotatable bond, here C and rotatable bond, here rotatable bond. And hydrogen bond acceptors, hydrogen bond donors. So likewise, when you are designing a molecule, you should follow typically the Lipinski rule of 5. And also these are the privileged structural elements. And it's like a structural toolbox when you have different heterocyclic compounds, 5-member, 6-member, mononitrogen containing, dinitrogen and trinatural nitrogen containing compounds. So these are some of the typical heterocyclic compounds, aromatic heterocyclic compounds, alicyclic heterocyclic compounds, and 6-member again 5-member. So these are the most of the heterocyclic compounds seen which are already in the market, the drugs are which are already in the market. So means these are act as like not only like a substitutions they are very good pharmacophores. Like they can act as a hydrogen bond donors and acceptors. So while designing molecules, you have to keep in mind, you have to introduce this type of pharmacophores in your molecule. And again bioisosteres. So this is introduced by Alice Federer in 1950s. Who is defined the compound eliciting similar biological effect. So for example, by introducing this bioisosterism in a molecule, so what happened? So it will increase improving potency, enhancing selectivity and altering physiochemical properties, reducing or deregulating metabolism and eliminating or modifying its oxidose and acquiring novel intellectual property. So for example, simple bioisosterism is simple. Hydrogen you replace with deuterium. Hydrogen you can replace with fluorine, or methyl CH3 you can replace with CF3. So what is the significance of this bioisosterism? So means it can improve the lipophilicity of the molecules and solubility. And for example, if you replace with CH3 with CF3, so it is like a very good lipophilicity molecule. Fluorine containing compounds are very good lipophilicity. So that's why most of these fluorine containing compounds they can easily cross the blood-brain barrier. BBB, blood-brain barrier. So that's why if you replace with proton with fluorine or CH3 with fluorine, so it can improve the lipophilicity of your designed molecules. So likewise, many further like phenols, catechals, carboxylic acids, amides. Amides you can replace with amide bond, peptide bond you can replace with triazole. And carboxylic acid functionality you can replace with tetrazole. These are like a bioisosteres. So these are like examples, very good example, monovalent versus trivalent and trivalent. So little bit I will give brief overview about cancer. So I will minimize in the introduction because already in the first hour he has given a very good introduction about cancer. So cancer is a uncontrolled cell proliferation, and again it is divided into two categories, that is benign tumor and malignant tumor. So benign tumor easily we can do surgery and even remove. But the problem is malignant cancer. So the malignant cancer can move from one part of the body to other part of the body through a systemic circulation. That is the main. So it is very difficult to cure malignant cancer, right? So here we are working on VEGFR inhibitors, vascular endothelial growth factor receptors. So these are like angiogenesis. What is meant by tumor angiogenesis? So angiogenesis is a formation of new blood cells, right? So they can create a microenvironment and supply oxygen and nutrient to the cancer cells, right? So in this VEGFR, vascular endothelial growth factor receptors play a very important role. So you can design small molecules, inhibit this VEGFR, so then you can disconnect that. So there is no supply of this nutrient and the oxygen to the cancer cells. So it is deemed to be, the cancer cells deemed to be killed. So cancer targets, if you see from 2012 to 2030, so worldwide cancer cases are projected 50% from 14 million to like 21 millions. And world cancer deaths are projected to 60% from 8 million to 13 millions. So these are the causes of cancer. 30% is tobacco and 20% cancers are like related to individual being obesity and overweight, and 16% of cancers are like bacteria and virus, and remaining are these one. So it's already explained about this one. These are the treatment for cancers, different hormone therapy, surgery, and bone marrow therapy, chemotherapy, targeted therapy, radiation therapy, and immunotherapy. So we are working mostly on chemotherapy. So these are the top 5 cancers in men and women. In men, lung cancer, and in women, breast cancer. If you see cancer today, this is total world population, you can count. So from that, number of new cases are finding almost 2 crores per annum, and number of deaths almost close to 1 crore. So number of related cases for 5 years is 5 crores. So cancer today, if you see, in 2022, 1 in 8, 12.9%, but if you see same 1975, 1 in 11, means 9 times. So from 9 to 12, 30% increase. And cancer tomorrow, so 2020, 19.3 millions, but if you go 2040, it is reaching to 28.9 million. So that's why the cancer discovery is they are investing, many of the companies a lot in the designs of new chemical entities with respect to cancer. So if you see the cancer drug revenue growth, its share of the pharma market, you see these are the 143.9 in 2019 total drugs, but in that 15.7% share is cancer drugs. If you see 2025, $282 billion in total market of drugs, but in farm only cancer $21.9 billion. So how much the cancer drug discovery and the pharma market is growing. So cancer drugs approved in the year 2020. Only year 2020, 19 drugs for different cancers they have been identified. And cancer drugs launched 2002 to 2021, how they increase exponentially, and globally 250 cancer drugs have been launched. And drugs in clinical trials for targeting cancer. These are all phase 1, phase 2 and phase 3 clinical trials. Most of the drugs. And most of the drugs are like combination drugs. So they are developing through the molecular radiation. So previously already established drugs are there. They are combining 2 drugs in a single entity to do multi-drug resistance through a molecular radiation approach. So these are the chemotherapeutic agents and its targets. So we are working mostly on tubulin proliferation inhibitors. So tubulin has a play a very important role in human. It is maintaining cell structure and intracellular transport and cell division. And again in tubulin, there are 2 types: tubulin proliferation inhibitors, tubulin deproliferation inhibitors. In tubulin perforation, weak alkalis and colchicine. In tubulin deperforation, taxanes and laminites. So this tubulin is having positive and alpha tubulin and beta tubulin, and is having positive and negative end. So these are the different binding site: microtubules, colchicine binding site, and then vinca site, and this side disturbing agents. This side is stabilizing agents. So we are working on tubulin perforation inhibitors or colchicine binding site. Next. So the marker, what is the concept of marker aggregation? So the structural modification tool for the aggregation of lead molecule with the ability to modulate more than one bioreceptor associated with the target disease. Okay, so this initial process consists identification of the marker recognition pattern of the pharmacophore, means they are known pharmacological properties, means they are well-known drugs. So for example, drug A and drug B, they are independently well studied. We know all the regulatory data, what are the mechanism of action, everything we know. So, and receptor A and receptor B. So here molecular aggregation is taking place between this A and B, like a hybrid and hybrid AB and a spacer, means linker based, merging based and fused based. So while molecular aggregation approach, you can take drug-like molecules and natural products and pharmacopores. You can do molecular aggregation, then you will get a new hybrid, new chemical entity. So that is like improved affinity, efficacy, effective modulation, and less side effects when compared to the parent drug molecules. So again in this process, component drug design plays very important role. So like structure based drug design and ligand based drug design. Structure based drug design is you know the three-dimensional structure of a protein or enzyme, and you can easily understand how you can design your molecule based on your target, based on the three-dimensional structure of the protein. And another way, ligand based drug design. So you don't need to know about the three-dimensional structure of the protein, but you know the drug molecule. So likewise, both approaches you can take, and you can do mixture screening, you can do millions of compound you can screen. From that you can identify a lead compound. So in this drug discovery process, by using component drug design, you can minimize some of the time. So these are the privileged scaffolds. As mentioned, the significance of natural products. That just now we had. So these are all natural products based molecules. So based on these natural products molecules, they have different mechanism of action. These molecules. So based on these molecules, we have similar kind of molecules. In synthetic way, we derive these molecules and we have synthesized. So again, this triazole molecules, this triazole 1, 2, 3 triazole is a primary retrocyclic compound. It's like a very good pharmacophore. As you know, this click chemistry, right? In 2022, so 2 years back, 3 professors received a Nobel Prize for this triazole molecule. So they discovered this click chemistry 2002, and after 20 years, the 3 of them received a Nobel Prize. So this click chemistry plays a very important role in all kinds of research. So this triazole molecule is available in the tazobactam, cefotaxime, and carboxy imidazole. These are having this triazole 5-member ring system. This is not only a pharmacophore, this is like a bridging motif, and it's like a connecting unit between 2 pharmacophores. So this can be easily synthesized by using click chemistry, like a azide, N3 and terminal alkyne. Simple copper catalyst. You can do 1,3 dipolar cyclation reaction. You can construct these beautiful 5-member tetracyclic compounds. So likewise, we have designed, synthesized a series of new chemical entities, and we have tested these molecules on selected human cancer cell lines and through the different mechanism of action. Some of the molecules are DNA minor group binders. So means they can selectively interact with the C2 amino group of the guanine base, where this you can see imine double bond. So this, for example, you see this imine double bond. So this selectively interact with C2 amino group of the guanine base and make it adaptive covalently. So that is like a mechanism of action. And some of the molecules are like a DNA intercalators, the sequence selectively they can interact with the DNA minor and major and some of them are through 5-5 starting interactions. So likewise, we have designed many molecules and they have shown very good anti-cancer activity on breast cancer cell lines, lung cancer cell lines and triple-negative breast cancer cell lines. So these are some of the building blocks. This is equals to solidity. Is a 3-alkynyl oxyindole. So it's marketed by Pfizer company. It's like a student. So it's like act as a tyrosine kinase inhibitor for the gastrointestinal tumor cancer. This is like a blockbuster drug. So but in this, if you see how we can design this molecule. If you cut here retrosynthetic, you will get this molecule oxidol, simple oxidol. How you can synthesize? Very simple from aniline. From aniline, you can do some reactions, chloral hydrate and concentrated, you will get this oxidol. Simple. Wolff-Kishner reduction. You all of you know, right? Wolff-Kishner. Here carbonyl compound reduction may take place, so then you will get active methylene group. So this is like a building block. Okay. How we utilize this oxidol molecule for the design of different molecules. And again, this is like a thiazolidinedione. This is like a PTR gamma receptor and very good cytotoxic agent. This is like a cancer drug. So we have taken this as one of the pharmacophore. So likewise, we have taken aldehyde and bromination and Suzuki cross coupling reaction for the CC1 formation, and we have taken same this oxidol, you can see. Oxidol with aldehyde, simple no-radical condensation reaction, you all of you know. Perkit condensation, no-radical condensation, right? So for the formation of this double bond, it's like a 3-alkynyl oxidol. So we prepared more than 20 compounds and we evaluated on selected human cancer cell lines, and 4 compounds have shown very good anti-cancer activity on prostate cancer. So likewise, again different enalkylated, different retrocyclic compounds. So these all retrocyclic compounds are available in the marketed drugs. So we have taken as a pharmacophore and same aldehyde we introduced and oxidol. 22 examples we have been prepared, and these one of the molecule has shown very good anti-cancer activity on prostate cancer cell lines, and the cell cycle arrest has been taken with G2M phase cell cycle, and these molecules have been apoptosis characteristic features we have found. We have done some staining studies, ethylene bromide staining, Dapi staining studies to see the morphology of these cells. How this cell distraction is taking place through a R-shaped shoe formation and cell doubling, chromatin condensation. So these are through staining studies we can observe when we treat it with our compound in a dose-dependent manner, how the cell distraction is taking place, how it is forming apoptic bodies. So these observations we have done. So likewise, so one of my student Ramya, she is working in Massachusetts Hospital, Harvard University, USA from last 2 years. So she derived, so these 22 hybrids on beta carbon indole alkaloids. So these are like indole alkaloids. So we have done simple novel condensation with aldehyde with oxindole. 22 derivatives we have done. And similarly, thiazolidinedione with the same again active methylene group with aldehyde. 27 hybrids we have been prepared. And we have done all in vitro conditions on different cancer cell lines and different staining studies we have seen, and we observe the cells how they are like destroying through. So different studies we have done. So these like this one of the molecules have shown on triple negative breast cancer cell line. So with 0.9 micromolar activity. So likewise, we have designed through molecular hybridization, you can see different pharmacophores like beta-carbolin, thiazolidinedione and indole. These all three we introduced in a one molecule, you can see, through thiazolidine formation and carboxamide. So this thiazolo, amido, bridged indole, beta-carbolin hybrids. So this is the synthetic way we have arrived, and we have synthesized a number of derivatives, and they have shown very good anti-cancer activity, and these some of the 2 molecules have shown very good interaction with the binding sites like compound 12C with the active packet of the 5G, GWK, and these are very good interrelative binding of 12C along with residual interactions also we found, and also we have calculated the binding energy. 12C is minus 58.91 and 12A is minus 53.67. So they are especially interacting with the different amino acids. And one more student, so he is Dr. Pankaj Sharma. He is my PhD student. He is now presently working second postdoc in Rutgers University, USA. So he has designed molecules, 25 examples like benzoyl molecules with thiazide through novel condensation reaction between active LIGO and RDI. So 25 examples we have been prepared, and these molecules have shown nanomolar activity on lung cancer cell lines. So a lot of biological studies we have done. I am not going to discuss these ones. So these molecules have been patented. The patent also been granted. And similarly, we have done a lot of derivatives of vegetables and thiazide derivatives. And one more student, Dr. Savani, she is working in School of Medicine, University of North Carolina, USA. So she has worked on pyrimidine linked molecular hybrids. This is like a simple pyrimidine molecule. We have constructed through the multi-compound reaction, Bigelow reaction, and we constructed this one. And different series of compounds, diversified compounds have been prepared. And these molecules act as a totally collaboration inhibitors. So these are like a cell cycles G2M phase and lots of mitochondrial membrane distraction, and we have done annexin binding service, and totally studies also we have done. So these molecules have shown very good anti-cancer activity. Even breast cancer, again dual electrolytic breast cancer. So again this pyrimidine based linker based one, thiourea and sulfur linkage. So these molecules have worked as like a vascular endothelial growth factor receptors. So these molecules have shown nanomolar activity, and these are like a urea based derivatives, 27 carbamide derived pyrimidine based conjugates. So these are shown very good anti-cancer activity on breast cancer cell lines. And primarily linker based pyrimidines. So we have been used this like as a linker. So pyrimidine based molecules. So these molecules have shown very good anti-cancer activity on different cancer like lung cancer, prostate cancer, breast cancer, and many of these cancer. These molecules not only tested on cancer cells, we also tested on normal cell lines. What is the safety of normal cell lines? So many folds these molecules are very safe to cancer cell lines. And through multi-component reactions, so we have done many of the compounds through multi-component reactions. So these are the natural products. So these are natural product type derivatives. We have synthesized through the multi-component reaction in a one-part manner. And these molecules have shown very good anti-cancer activity on selected skin cancer cell lines, these are lung cancer cell lines, and prostate cancer cell lines. And these are like a fused system through intramolecular recombinant dipole cyclation. So these 5 compounds have been prepared, 20 compounds, and these tetrazoal compounds have been prepared, and these are also shown very good anti-cancer activity. So these are like linker based. So these beta carbonyl and carbazamide. So these are like DNA interplators. These are like a totally collaboration inhibitors, collagen binding sites. So these both we have connected to the amide linkage. So like a linker based. Yeah. So these are like molecular aggregation. Different pharmacophores we have been taken, and molecular aggregation has been taken place. We got these all pharmacophores in a single entity, and we have done some dogging studies, simulation studies, and we have seen how they are interacting and what is the essential of this cis conformation and how it is like a interacting with toluene binding site. Likewise, we have designed so many nuclear entities, and these are some of the potent compounds which are patented and patents also granted. And we are not only working on design system capabilities, and also we are working in the development of novel methodologies, sustainable methodologies, and for the CH activation, green chemistry, and combinable chemistry, microwave assisted reactions. So these are some of our notable. And also we have contributed some of the innovative these are like diethylcarbonates. Those are this one. And these are like spiral compounds. These are these micro-resistive reactions for the 3-member to 6-member reactions. And again, spiral compounds like azide-based molecules. This revealing the polyheterocyclic compounds by micro techniques for all polyheterocyclic compounds. And this unveiling KM4 histodemethylase inhibitors in cancer therapy. This is published in the Discovery today. And these sulfur-containing compounds, regular sulfur-containing compounds in heterocyclic and especially in anti-cancer and their update. And this is like a multi-compound reactions and the mechanism insights published in OCF. And role of sulfamides and CH activation reactions. And this transition methyl CH activation reactions. And exposure studies in construction of 2-oral benzoxazole and disease review. And recent evidence on accelerated circulation approaches and some other micro reactions. So in summary, so we have involved, so the reason we have done more than 2000 nuclear militaries we have synthesized in our laboratory, and we have tested those molecules are selected human cancer cell lines. Some of the molecules have shown non-molar activity, those molecules we have patented, and we efficiently utilize the chemistry, triazole as a best linker. And our all starting materials are very like cheaper, cost-effective starting materials have been utilized, and most of the molecules and the molecular weight is below 600. And the most fundamental lasting object of this is not production of new compounds, but production of properties. As a medicinal chemist, how we can introduce here functionalities like OH and LH, and how we can increase the lipophilicity of the molecules. And the right science and the right technology that are available could be utilized in the right manner, at the right time, and to develop right products for the right patients. So these are my students who they have completed their PhDs, and presently these students are working, 11 PhDs and 1 postdoc working, and these are the master students they are working for their dissertation work. So these are some of the students, past and present students. And so I must thank all my students. Without their involvement, without their range work, it is very difficult to achieve this much. And also I must thank my collaborators. He is my postdoc boss, Raman Kamal, and he is my PhD guide, and he is my postdoc boss. And I also worked in Brazil for some time with Ronald Alaspilli and Marcus Ederling. And also the Suresh K. Bhagava is Army Australia, who has carried out some of the biological work. And Narendra Nagesh from CCM is a scientist. He recently retired. We worked with many of the biological studies. And Dr. Satish, my student, is working in Chile. And Dr. Chandraya, Dr. Manoj Bandekar, Dr. Santosh Guru. These are the pharmacology faculty in Hyderabad. They are supporting a lot for the biological activity. Also, I must thank the ICMR as well as the DST and Department of Pharmaceutical Institute of Chemicals and Fertilizers for supporting financial support to carry out this entire research work. So thank you.
Thank you so much, sir. Now the session is open for question and answer. If anybody is having question, please stand up.
That means previously you told that nearly 3 to 5% cancer problem is there in the population for each and every 10 to 20 years. Can you suggest what are the precautions, what we have to take to reduce that?
As I mentioned, more than 30% cancer cases are by consuming tobacco, cigarette. Okay, only 5%, 5 to 10% are genetic. So means that we can't change because it's like a parental.
Yes, sir.
Okay, but remaining 90% cancers are based on our habits.
Yes, sir.
Okay, we are polluting our environment. Okay, and our lifestyle, physical activity, okay, diet. So these all depending upon. So if you change those habits, you can avoid.
Yes, sir. Thank you.
And as I wish, the morning also there are so many questions came across the students and faculty for about cancer and their side effects and their targeted approach. See, every drug is a side effect. Even if you take food also more than sufficient, that is also poison. Right or wrong? Okay, so anything is there, drugs are more side effects are there is no selective because it's going to kill cytotoxicity means it's going to cancer cells means it also kills normal cells also. So cancer cell because when you are taking drug it will go and metabolize, right? It is distributed to all parts of the body. It will not go selectively to the cancer cells. So though they are overexpressed, but there are lot of side effects for cancer. Okay, so that's why it's very important. Our lifestyle changes are very, very important.
Yes, sir. Myself, Dr. N. sir. Sir, after pandemic, we have in the market we have a branded, generic and counterfeit spurious drugs. Pardon? In the market we have a branded drugs, generic drugs, and also we have nowadays after pandemic we have a counterfeit, spurious, fake drugs are also available. Okay, how we can determine or predict the drug is like a spurious or fake or branded or generic?
So that is like a drug control, their role, isn't it?
Yes, as a pharmacy professions, could you suggest anything, sir?
So that's why they have to promptly, they have to take the sample and they have to check.
That's it.
That's why they have to check.
Okay. It's look like say
the Suresh mentioned, even in herbals also other is taking place. So what they will take natural extract. So they will diclofenac powder, they will mix it and they will give automatically they will go.
Yes.
So automatically patient will go. So but when you will check that whatever it is herbal or whatever if you check through mass spectrometry, then you will find whether they are adding diclofenac or not or how much natural it is, you can identify.
Yes, sir. As a common man, they don't know about these things.
Obviously,
yes, sir. That's
why we need data to solve the
Yes, sir. Thank you.
Good afternoon, sir. Good afternoon, sir.
Yeah.
So my question was, what can be the possible complications in pediatric and geriatric patients, and how we can overcome it?
Pardon?
What can be the possible complications in pediatric and geriatric patients, and how we can overcome it?
So I am not understanding.
I'm asking about the complications, possible complications which we come across in like age differences of patients.
Okay. By using these drugs.
Yeah.
Okay. So I am not aware about the detail about that.
Oh.
Yeah. Sorry.
Anybody having question?
Good afternoon, sir. Sir, camptothecin is anti-cancer drug.
Yes.
Sir, Nothopodytes pneumonia is plant major source. Narcia is basically found in Western Ghats. Actually, it is.
I know about camptothecin is a topoisomerase inhibitor.
Yes, sir. But I have, sir, it is enlisted in endangered plant. Government endangered list. Sir, if we can cultivation purpose, the government can provide us this funding or.
I think Dr. Suresh Babu can answer this one. It's a natural product.
Government is encouraging the farmers to cultivate the medicinal plants. There is a medicinal and aromatic plant board is there in the Ministry of Ayush. So they will buy the plants.
Thank you, sir.
Thank you so much, sir. Now I conclude that question answer session is over. I would like to say thank you to Dr. Shankaraya sir for the to providing us immense knowledge about these anti-cancer therapies. It was a very nice lecture. Please give a big round of applause to him. Now I request Dr. Vishal Babar sir to felicitate Dr. Swapnil Fade sir, Principal of College of Pharmacy, Panu. much, sir. Now I declare that the participants will have a lunch break up to 2:30 p.m. After exactly at 2:30 p.m. we will have our valedictory session and prize distribution. So all participants please present at 2.
कस काय बरं एंड करू का मग सेक्शन एंड स्ट्रीम करू का एंड कर तू ते फोन घेऊच नाही करू का काय म्हणत आहे करू का एंड मग इथली हा तू एंड जाऊ दे ओके थांबला आता हे